Hello and welcome. I'm truly delighted you're here. Before we get underway, I'd love for you to share in the comments where you're tuning in from and what time it happens to be in your part of the world. It's always such a joy to see this community come together across places and time zones. I also want to take a gentle moment to thank everyone who's subscribed and shown support through likes on the videos. It helps me know what you value most, and it helps others find this space too. Please feel free to leave your ideas for future topics in the comments. Now, let's begin. Chapter 1: Understanding Enzymes as Biological Catalysts. Enzymes are remarkable molecules that serve as biological catalysts, playing a pivotal role in facilitating the countless chemical reactions that sustain life. Imagine a world without enzymes. Processes that occur within us, such as digestion, energy production, and even the replication of our genetic material, would come to a near standstill. Enzymes are essential for life, acting as accelerators that lower the energy barriers required for reactions to proceed. They are nature's catalysts, speeding up reactions without being consumed in the process. To understand enzymes better, visualize them as intricate machines operating within the cells of our body. Each enzyme possesses a unique structure, tailored to its specific function. This structure typically includes an active site, a specially shaped pocket that binds to substrates, the reactants in the reactions that the enzyme catalyzes. The binding of a substrate to an enzyme is akin to a key fitting into a lock, a perfect match that allows the enzyme to facilitate the transformation of the substrate into a product. The specificity of enzymes is one of their most fascinating attributes. Each enzyme is designed to catalyze a particular reaction or set of reactions. This specificity arises from the precise arrangement of amino acids that make up the enzyme's structure. Consider the words of the renowned biochemist, Arthur Kornberg, who stated, "Enzymes are the workers of the cell. Their ability to perform specific tasks efficiently is crucial to maintaining the balance of biological processes." Enzymes are not static entities. They undergo conformational changes upon substrate binding, an adjustment often referred to as the induced fit model. This model illustrates how enzymes can change shape to accommodate their substrates better. Envision a glove that molds itself to fit the contours of a hand perfectly. This flexibility enhances the enzyme's ability to catalyze reactions effectively, ensuring that metabolic pathways can proceed smoothly. The importance of enzymes in metabolism cannot be overstated. Metabolism encompasses all the chemical reactions that occur within a living organism, and enzymes are the driving force behind these reactions. For instance, consider the breakdown of glucose during cellular respiration. In this intricate process, enzymes like hexokinase and phosphofructokinase play vital roles, enabling our cells to extract energy from nutrients. Without these enzymes, the energy stored in glucose would remain locked away, inaccessible to our cells. Enzymes also facilitate anabolic reactions, where smaller molecules are combined to form larger, more complex ones. An example of this is the synthesis of proteins from amino acids. The enzyme ribosome catalyzes this process, ensuring that proteins are constructed with precision and efficiency. It is through these anabolic and catabolic pathways that cells maintain homeostasis, the delicate balance necessary for life. The impact of enzymes extends beyond individual cells. They are central to the functioning of entire ecosystems. In nature, enzymes break down organic matter, allowing nutrients to be recycled and made available to other organisms. The work of enzymes in decomposing dead plants and animals enriches the soil, promoting plant growth and sustaining life. This interconnectedness highlights the importance of enzymes not just in isolated biological processes, but in the grand tapestry of life itself. Moreover, enzymes have been harnessed by humans for various applications, from medicine to industry. The discovery of penicillin, for instance, revolutionized medicine, thanks in part to the enzyme-producing fungus that enabled its creation. Enzymes are also utilized in the production of biofuels, detergents, and food processing, showcasing their versatility and significance in our daily lives. The study of enzymes has not only enhanced our understanding of biological processes, but has also led to groundbreaking advancements in scientific research. One such outstanding figure in this field is Emil Fischer, who was awarded the Nobel Prize for his work on enzyme specificity. He proposed the lock-and-key model of enzyme action, a concept that simplified our understanding of how enzymes interact with their substrates. Fischer's insights laid the foundation for future research, allowing scientists to delve deeper into the world of enzymology. As we explore the intricate world of enzymes, we encounter a multitude of fascinating questions. What happens when enzymes malfunction? The repercussions can be dire, leading to various diseases and disorders. For instance, lactose intolerance arises from the insufficient production of lactase, the enzyme responsible for breaking down lactose in dairy products. Imagine the discomfort experienced by those who are unable to properly digest certain foods, a reminder of the crucial role enzymes play in our daily lives. Additionally, the study of enzymes has opened avenues for biotechnology, where scientists engineer enzymes to improve their efficiency or alter their specificity. This process, known as enzyme engineering, holds great promise in addressing various challenges, from developing sustainable energy sources to creating more effective pharmaceuticals. The potential for innovation is vast, and the journey into the world of enzymes continues to unfold. Enzymes also exemplify the remarkable precision of nature, showcasing how evolutionary processes have fine-tuned these catalysts over millions of years. The diversity of enzymes found in various organisms highlights their adaptability to different environments and metabolic needs. From the heat-loving enzymes of thermophilic bacteria to the cold-active enzymes of polar organisms, each enzyme reflects the unique conditions of its origin. Imagine the beauty of enzymes as they have evolved, each one a testament to the resilience of life. As biochemist Richard Dawkins once remarked, "We are survival machines, robot vehicles blindly programmed to preserve the selfish molecules known as genes." Enzymes, in their relentless pursuit of catalyzing reactions, embody this essence of life, tirelessly working to ensure the survival and thriving of organisms. As we journey further into the realm of enzymes, we begin to appreciate the elegance of their interactions at a molecular level. Enzyme kinetics, the study of the rates at which enzymes catalyze reactions, reveals insights into the efficiency of these biological catalysts. Factors such as temperature, pH, and substrate concentration can significantly influence enzyme activity. For instance, each enzyme has an optimal temperature and pH at which it functions best, a sweet spot that allows it to operate with maximum efficiency. Consider the delicate balance of conditions necessary for enzymes to perform their roles. Too much heat can denature an enzyme, altering its structure and rendering it inactive, while extreme pH levels can disrupt the interactions that maintain its shape. This fragility speaks to the precision required for life to flourish, as enzymes operate within tightly regulated environments to ensure the smooth functioning of biological processes. The exploration of enzymes also leads us to the realm of inhibitors and activators. These molecules can either reduce or enhance enzyme activity, providing another layer of regulation within metabolic pathways. Competitive inhibitors mimic substrates, competing for the active site of an enzyme, while non-competitive inhibitors bind to an enzyme elsewhere, affecting its function without competing for the active site. This intricate dance of molecules showcases the complexity of biological systems, where regulation is key to maintaining equilibrium. Imagine the interplay of enzymes, substrates, and inhibitors as a finely tuned orchestra, each component contributing to the harmony of life. This orchestration allows organisms to adapt to changing environments, ensuring that life continues to thrive in the face of challenges. As we reflect on the profound implications of enzymes, we begin to understand their role as messengers of life. They are not mere catalysts, they are the embodiment of biological processes that shape our existence. Enzymes allow for the conversion of energy, the synthesis of essential molecules, and the regulation of metabolic pathways, all of which contribute to the intricate web of life. In contemplating the future of enzymology, we are reminded of the endless possibilities that lie ahead. As scientific knowledge expands, so too does our understanding of these remarkable catalysts. The potential for new discoveries, applications, and innovations is boundless, promising to enhance our lives in ways we may yet only dream of. As we draw this exploration of enzymes to a close, consider the world of possibilities they present. Each enzyme is a key to unlocking the mysteries of life, a small yet powerful force that drives the processes essential for survival. The story of enzymes is one of precision, adaptability, and resilience, a narrative woven into the very fabric of existence. As we continue to uncover the intricate world of enzymes, we can delve deeper into the concept of enzyme specificity and how it shapes the very fabric of metabolic pathways. Each enzyme's unique structure is not merely a random arrangement of amino acids. It is a finely tuned configuration that has evolved to interact with specific substrates. This level of specificity ensures that biological reactions occur with remarkable precision, allowing for the complex interplay of metabolic processes that sustain life. Imagine a delicate balance where each enzyme interacts with its substrate as a musician would with their instrument, producing a symphony of biochemical reactions. This specificity is vital not only for efficiency, but also for the regulation of metabolic pathways. The phenomenon known as allosteric regulation further exemplifies this adaptability. Allosteric enzymes possess sites other than the active site, where molecules can bind and influence the enzyme's activity. This mechanism allows cells to finely tune their metabolic processes in response to changing conditions, a beautiful dance of regulation that ensures survival in fluctuating environments. In the realm of human health, the implications of enzyme specificity and regulation are profound. Consider the role of enzymes in pharmaceuticals. Many drugs are designed to mimic substrates or inhibitors, targeting specific enzymes to alter their activity. The development of statins, for example, showcases this principle. These drugs inhibit an enzyme involved in cholesterol synthesis, effectively lowering cholesterol levels and reducing the risk of heart disease. This targeted approach underscores how a deeper understanding of enzymes can lead to innovative treatments, transforming the landscape of medicine. The story of enzymes also leads us to explore enzyme cofactors and coenzymes, essential partners that assist enzymes in their catalytic activities. Cofactors can be ions or small molecules that bind to enzymes, often transforming them into an active form capable of catalyzing reactions. Coenzymes, on the other hand, are organic molecules that participate in the enzymatic reaction, often acting as carriers of electrons or functional groups. For instance, the coenzyme NAD+ plays a crucial role in cellular respiration by facilitating the transfer of electrons during metabolic reactions. These intricate relationships highlight how enzymes do not work in isolation. They are part of a larger network of interactions that propel biochemical processes forward. As we contemplate these partnerships, we may wonder about the many pathways of life and how enzymes fit within them. Consider the vast array of metabolic pathways that exist, each governed by a series of enzymes working harmoniously. Glycolysis, the citric acid cycle, and oxidative phosphorylation are just a few examples of the interconnected processes that rely on enzymes to convert substrates into energy, showcasing the elegance of biological systems. Picture a bustling city, with enzymes as the traffic signals, directing the flow of biochemical reactions and ensuring that everything runs smoothly. The exploration of enzyme evolution offers yet another layer of intrigue. Enzymes are not static. They evolve in response to environmental pressures and the needs of the organisms that utilize them. The concept of enzyme evolution allows us to appreciate the adaptability of life itself. For example, the enzyme lactase, responsible for breaking down lactose, has evolved in some human populations in response to dairy consumption. This adaptation illustrates how enzymes can reflect the relationship between organisms and their environments, a testament to the power of natural selection. As we turn our gaze to the future, one cannot help but feel a sense of wonder at the possibilities that lie ahead in the study of enzymes. The field of synthetic biology is emerging, where scientists are engineering enzymes to perform novel functions, paving the way for groundbreaking applications in medicine, environmental science, and biotechnology. Imagine a world where enzymes are custom designed to break down pollutants or produce sustainable biofuels, transforming our approach to pressing global challenges. The potential for innovation is as vast as it is exciting. In this journey through the world of enzymes, we have touched on their roles as biological catalysts, their intricate structures, and their profound impact on metabolism and life processes. Each facet of enzymes we explore reveals a deeper understanding of the complexity and beauty of life at the molecular level. As we continue to study these remarkable molecules, we are reminded that they are not merely catalysts. They are key players in the ongoing story of life, guiding and facilitating the processes that sustain us all. Chapter 2: How Enzymes Speed Up Reactions. Enzymes, with their remarkable ability to accelerate chemical reactions, operate through intricate mechanisms that involve lowering activation energy. This concept is fundamental in understanding how enzymes facilitate the myriad reactions that occur within living organisms. Activation energy can be thought of as a hurdle that must be overcome for a reaction to proceed. Imagine a boulder at the top of a hill, requiring a certain amount of energy to roll down the other side. Enzymes act as gentle guides, providing the necessary assistance to help that boulder find its way down with far less energy than would be required without them. When an enzyme binds to its substrate, it forms what is known as an enzyme-substrate complex. You can visualize this relationship as a dance, where the enzyme and substrate move together in harmony. This complex is pivotal because it stabilizes the transition state of the reaction, which is a temporary and unstable state that occurs during the transformation of substrates into products. By lowering the energy needed to reach this state, enzymes dramatically speed up reactions, allowing life-sustaining processes to occur efficiently. One of the most notable aspects of enzymes is their specificity, which hinges on the precise interaction between the enzyme and its substrate. This interaction can be likened to a snug fit, where only the correct substrate can bind effectively to the enzyme's active site. As the renowned scientist Daniel Koshland once said, enzymes are not merely catalysts, they are highly specialized machines. This specialization is what allows enzymes to facilitate specific reactions, ensuring that the complex biochemistry of life runs smoothly. To further appreciate how enzymes operate, let us consider the factors that influence their activity. Temperature plays a crucial role in enzyme function. Each enzyme has an optimal temperature range within which it operates most efficiently, much like how a flower thrives best in a specific season. At too low a temperature, molecular movement slows down, reducing the likelihood of enzyme-substrate collisions. Conversely, when temperatures rise beyond the enzyme's optimal range, the structure of the enzyme can become distorted. This phenomenon, known as denaturation, results in a loss of function. Imagine the warmth of the sun on a cool day, energizing the world around you. In the same way, moderate increases in temperature can enhance enzyme activity, leading to a faster reaction rate. Yet, the moment that warmth becomes too intense, the delicate balance shifts, and the once efficient enzyme may falter. Another significant factor influencing enzyme activity is pH. Enzymes, like delicate flowers, have their own preferences when it comes to acidity or alkalinity. Each enzyme has an optimal pH level at which it performs best. When the pH strays too far from this optimal range, the enzyme's structure, including its active site, may also undergo changes. This alteration can interfere with its ability to bind substrates effectively. Consider the enzymes in your stomach, which thrive in a highly acidic environment, aiding in the digestion of food. Their efficiency is a marvel of biological design. Now, picture enzymes that function in the more neutral environment of the bloodstream, illustrating the diverse adaptability of these biological catalysts. As we delve into the intricacies of enzyme activity, it becomes evident that the world of enzymes is governed by a delicate balance of conditions. Factors such as substrate concentration, temperature, and pH interact in complex ways to determine the speed and efficiency of biochemical reactions. This intricate interplay ensures that our bodies can respond to changing conditions and maintain homeostasis. The exploration of enzyme function has been enriched by the insights of many great scientists. One such figure is Emil Fischer, who proposed the lock and key model to describe how enzymes and substrates interact. His vision helped lay the groundwork for our understanding of enzyme specificity and catalysis. Fischer's work was not merely theoretical. It was grounded in meticulous experimentation and observation, demonstrating a profound connection between scientific inquiry and the understanding of life. Imagine the feeling of uncovering a hidden treasure, each layer revealing more about the intricate workings of life itself. The study of enzymes is much like that. Every discovery adds depth to our understanding of biological processes. The realization that enzymes are not static entities, but dynamic participants in a symphony of reactions, highlights their significance in the grand design of life. As we continue this exploration, we can appreciate that enzymes are not just passive helpers, but active players that respond to the environment around them. Their ability to adapt to varying conditions speaks to the resilience of life itself. In the heart of every cell, enzymes tirelessly work to maintain the delicate balance necessary for survival, ensuring that energy production, nutrient breakdown, and many other essential functions proceed smoothly. In summary, the mechanisms by which enzymes lower activation energy, the formation of enzyme-substrate complexes, and the factors influencing enzyme activity create a fascinating narrative of life at the molecular level. Through the lens of scientific inquiry, we uncover the extraordinary capabilities of these biological catalysts, illuminating the pathways that sustain life. Imagine each enzyme as a tiny hero, tirelessly working behind the scenes, ensuring that the intricate processes of life unfold effortlessly and beautifully. With this understanding, we can appreciate not only the importance of enzymes, but also the wonder of the natural world, where every reaction is a testament to the elegance of biological design. As we reflect on these insights, we begin to see that the world of enzymes is an invitation to marvel at the complexity and beauty of life itself. As we journey further into the realm of enzymes, we can explore the dynamic nature of enzyme-substrate interactions. Enzymes are not static structures; they exhibit remarkable flexibility. This adaptability allows them to adjust their conformation in response to substrate binding, a phenomenon known as induced fit. Imagine a glove that molds itself to the hand of the wearer, providing a snug and comfortable fit. This flexibility enhances the enzyme's ability to catalyze reactions efficiently, ensuring that the substrate is perfectly positioned within the active site. This concept of induced fit shows that the interaction between an enzyme and its substrate is not just a simple lock-and-key mechanism, but a more intricate and responsive relationship. The enzyme undergoes subtle changes upon binding, stabilizing the transition state and facilitating the conversion of substrates into products. This underscores the elegance of enzyme action, where even the slightest molecular adjustments can lead to profound outcomes in biochemical processes. Moreover, the formation of enzyme-substrate complexes is crucial for understanding the speed at which reactions occur. After the enzyme binds to its substrate, the reaction can proceed with remarkable rapidity. Enzymes can operate at astonishing rates, transforming substrates into products in a fraction of a second. This efficiency is vital in biological systems, where timely reactions are essential for maintaining life. Consider the role of enzymes in metabolic pathways, where a cascade of reactions must occur in a coordinated manner. Each enzyme acts as a facilitator, ensuring that substrates are converted into products without delay, allowing the organism to respond to its environment. The intricate choreography of these enzymes exemplifies the beauty of biological systems, where chemistry meets life in a seamless flow. The influence of temperature and pH on enzyme activity reveals yet another layer of complexity. Each enzyme has its unique profile of optimal conditions, tailored to its specific environment. For instance, enzymes in thermophilic organisms, which thrive in extreme heat, have evolved to maintain their structure and function at elevated temperatures. Imagine the resilience of life as it adapts to the most inhospitable environments, showcasing the remarkable innovation of evolution. Conversely, enzymes from organisms living in cooler climates display stability and efficiency under lower temperatures. This adaptability not only highlights the diversity of life, but also emphasizes the importance of enzymes in various ecological niches. The ability of enzymes to function across a range of temperatures and pH levels is a testament to their evolutionary significance and their role in sustaining life. The delicate balance of enzyme activity also extends to the concept of enzyme inhibitors, substances that can slow down or halt enzyme function. Inhibitors can be competitive, binding to the active site and blocking substrate access, or non-competitive, binding elsewhere on the enzyme and altering its activity. This interplay between enzymes and inhibitors is crucial in regulating metabolic pathways, ensuring that reactions occur at the right time and place. Enzyme inhibition is not merely a hindrance. It can also be harnessed for therapeutic purposes. The development of drugs that target specific enzymes has revolutionized medicine, allowing for the treatment of various diseases. Consider the story of penicillin, discovered by Alexander Fleming. This antibiotic works by inhibiting bacterial enzymes, disrupting their ability to form cell walls, and ultimately leading to their demise. Such breakthroughs illustrate how understanding enzymes can lead to innovations that transform our approach to health and disease. As we reflect on the various factors that influence enzyme activity, we must also acknowledge the role of substrate concentration. The relationship between substrate concentration and reaction rate is often depicted as a curve, illustrating how, at lower concentrations, increases in substrate lead to proportionate increases in reaction rates. However, as saturation occurs, the reaction rate approaches a maximum, where all available enzyme active sites are occupied. Imagine a busy marketplace where vendors are selling their goods. Initially, as more customers arrive, sales increase steadily. However, once every vendor is engaged with a customer, the pace of transactions cannot increase further, regardless of how many more customers appear. This analogy captures the saturation effect seen in enzyme kinetics, where the availability of enzymes ultimately limits the reaction rate, highlighting the importance of enzyme concentration in biochemical processes. The extraordinary capabilities of enzymes empower biological systems, driving the essential reactions that sustain life. The stories of individuals who have contributed to our understanding of enzymes are as inspiring as the enzymes themselves. For instance, the work of Frederick Sanger, who developed sequencing techniques that revealed the structure of enzymes at an atomic level, deepened our appreciation for these remarkable molecules. His insights allowed us to comprehend not only how enzymes function, but also how their failure can lead to disease. As we continue to explore the world of enzymes, we find ourselves drawn into a narrative that transcends mere chemistry. The interplay of enzymes, substrates, and the myriad factors influencing their activity paints a vivid picture of life at the molecular level. Each reaction catalyzed by enzymes is a step in the intricate dance of existence, where every moment is a testament to the complexity and beauty of life. Imagine standing at the edge of a vast forest, observing the myriad interactions of life within. Each enzyme is like a tiny creature, playing a role in the grand ecosystem of biochemical reactions that support the flourishing of life. This intricate web of interactions is not only fascinating, but also essential for our survival, reminding us of the interconnectedness that characterizes all living organisms. As we delve deeper into this exploration of enzymes, we can appreciate the challenges they face and the adaptations they have undergone throughout evolution. From the depths of the ocean to the peaks of mountains, enzymes have evolved to thrive in diverse conditions, showcasing the remarkable resilience and ingenuity of nature. This adaptation is not merely a survival mechanism, but a testament to the extraordinary journey of life on Earth. In this ongoing story of enzymes, we find an invitation to marvel at the incredible intricacies of life itself. Each discovery adds another layer of understanding, illuminating the pathways through which life unfolds. The elegance of enzyme action serves as a reminder of the delicate balance that sustains us, allowing for the beautiful complexity of existence to continue. Chapter 3: Enzyme Specificity and Affinity. In the intricate world of biochemistry, the relationship between enzymes and their substrates is defined by a remarkable specificity, akin to a perfectly shaped key fitting into a lock. This idea, known as the lock and key model, illustrates how each enzyme is uniquely suited to catalyze a particular reaction. Picture a vast array of keys, each crafted with precision, ready to unlock specific doors, representing the diverse substrates that enzymes interact with. This model emphasizes the rigid structure of enzymes, suggesting that their active sites are perfectly molded to fit their corresponding substrates. However, this concept is merely the beginning of understanding enzyme behavior. The beauty of life's chemistry lies not only in the precision of the lock-and-key fit, but also in the subtle flexibility that enzymes exhibit. This flexibility is encapsulated in the induced fit model, which suggests that when a substrate approaches an enzyme, the active site undergoes a conformational change to better accommodate the substrate. Imagine a warm embrace where the arms of one person adjust to hold another comfortably. This dynamic adaptation allows for a more efficient interaction, enhancing the enzyme's ability to catalyze reactions. Consider the implications of these models in the context of metabolic pathways, the series of interconnected reactions that sustain life. Each enzyme operates within a larger network, ensuring that metabolic processes proceed smoothly and efficiently. The specificity of enzymes is paramount here. It ensures that each reaction is carefully regulated, preventing chaos within the cell. Imagine the delicate balance of a finely tuned orchestra, where each musician plays in harmony, their roles clearly defined. The significance of enzyme specificity can be illustrated through the work of Emil Fischer, who proposed the lock-and-key model over a century ago. His insights laid the groundwork for future discoveries, shaping our understanding of enzymatic function. Fischer once remarked, "The specificity of an enzyme is like that of a key to a lock." This perspective highlights the inherent design of enzymes, providing a framework for their catalytic activities. As we reflect on the metabolic pathways in which these enzymes operate, we can envision the pathways as intricate roads, each leading to a specific destination. The enzymes act as traffic lights, guiding molecular traffic to ensure that reactions occur in a controlled and efficient manner. This specificity prevents unwanted side reactions that could lead to detrimental effects on cellular processes. Moreover, the role of enzymes extends beyond mere catalysis. They are also critical in maintaining homeostasis within the body. For instance, enzymes involved in glucose metabolism demonstrate how specificity is vital for energy regulation. When blood sugar levels rise, enzymes such as hexokinase catalyze the phosphorylation of glucose, converting it into glucose 6-phosphate, thus facilitating its storage or utilization. This process is tightly regulated, ensuring that the body maintains an optimal energy balance. The induced fit model further illustrates how enzymes can fine-tune their interactions with substrates, allowing for variations in the active site that can adapt to different substrates. This adaptability is crucial in environments where substrates may vary due to dietary changes or metabolic demands. It is a testament to the resilience and versatility of life at the molecular level. To appreciate the elegance of enzyme specificity, one must also consider the evolutionary perspective. Enzymes have evolved over billions of years, honing their specificity to meet the demands of an ever-changing environment. The concept of evolution by natural selection suggests that those enzymes which exhibit greater specificity for their substrates are more likely to succeed in facilitating essential reactions, passing their advantageous traits to future generations. This evolutionary journey has produced enzymes with remarkable capabilities. Take, for example, the enzyme lactase. In humans, lactase facilitates the breakdown of lactose, the sugar found in milk. In some populations, the ability to digest lactose into adulthood is a result of a genetic adaptation that arose in response to dietary changes. This specificity is not merely a biochemical curiosity, but a reflection of human history and adaptation. The specificity of enzymes also has practical implications in biotechnology and medicine. Enzymes are harnessed in various applications, from industrial processes to pharmaceuticals. For instance, the enzyme amylase, which breaks down starch into sugars, is widely used in the food industry for producing sweeteners and in brewing. Its specificity allows for efficient results, minimizing unwanted byproducts. Similarly, in medicine, enzymes play a crucial role in diagnostics. Enzyme-linked immunosorbent assays, or ELISAs, utilize the specificity of enzymes to detect the presence of specific antibodies or antigens in a sample. This technique has revolutionized disease detection, allowing for rapid and accurate diagnoses. As we ponder these applications, we can ask ourselves, how does the specificity of enzymes impact our daily lives? The answer resonates in the food we consume, the medications we take, and the very processes that sustain our existence. Enzymes are not just biological catalysts, they are integral to the fabric of life. In conclusion, the intricate relationship between enzyme specificity and affinity is a testament to the complexity of life at the molecular level. From the lock-hand key and induced fit models to the evolutionary adaptations that have shaped these remarkable molecules, the story of enzymes is one of precision, flexibility, and profound impact on the biological world. As we continue to explore this fascinating realm, we uncover not only the mechanisms of life, but also the interconnectedness that binds all living things together. The lock-hand key and induced fit models beautifully illustrate the fundamental principles of enzyme specificity, yet they are merely the surface of a much deeper understanding of how enzymes operate within the intricate web of biological systems. Enzymes must not only recognize their specific substrates, but also respond to the cellular environment's dynamic conditions. This responsiveness is essential for the regulation of metabolic pathways, ensuring that energy production, nutrient processing, and biosynthesis occur in harmony. Imagine a bustling marketplace where vendors sell various goods, each tailored to meet specific needs. In this metaphor, enzymes act as specialized vendors, each offering a unique product suited to a particular customer, representing substrates. The marketplace thrives on the interactions between these vendors and customers, much like a cell thrives on the interactions between enzymes and substrates. This vivid imagery encapsulates the essence of how enzyme specificity allows for finely tuned metabolic regulation. The importance of this specificity becomes even more apparent when we consider the consequences of a mismatch. Enzymes that fail to bind to their intended substrates may catalyze unwanted reactions, leading to the accumulation of toxic byproducts or the depletion of essential metabolites. This disruption can significantly impact cellular homeostasis, highlighting the need for precise control within metabolic pathways. An exemplary instance of the consequences of enzyme specificity is found in the realm of metabolic disorders. Phenylketonuria, a genetic condition, arises from a deficiency in the enzyme phenylalanine hydroxylase. This enzyme's role is to convert phenylalanine, an amino acid found in many protein-containing foods, into tyrosine. Without this specific enzymatic action, phenylalanine accumulates to toxic levels, leading to severe neurological issues. The delicate balance maintained by enzymes underscores their critical role in health and disease. As we explore further, let us consider the flexibility inherent in enzyme-substrate interactions. The induced fit model, with its emphasis on adaptability, reveals that enzymes can accommodate slight variations in their substrates. This adaptability is particularly important in a biological context where conditions and substrates may vary. Enzymes can adjust their active sites to optimize binding, much like a craftsman who adjusts tools to fit different materials. This flexibility can be observed in the enzyme chymotrypsin, a digestive enzyme that breaks down proteins. Chymotrypsin exhibits remarkable specificity for certain amino acids, yet it can also adapt to accommodate various peptide substrates. This duality allows for effective digestion while maintaining the precision necessary to prevent the breakdown of beneficial proteins. The ability to adjust while remaining true to their function illustrates the fine balance enzymes maintain. The fascinating interplay between enzyme specificity and environmental factors extends beyond mere adaptability. Enzymes are influenced by temperature, pH, and the presence of inhibitors or activators. These factors can dramatically alter enzyme activity, leading to shifts in metabolic pathways. For instance, a slight increase in temperature can enhance enzymatic activity up to a certain point, after which the enzyme may denature and lose its function. This delicate equilibrium is akin to a finely tuned instrument, where even the smallest adjustments can create a symphony or discord. The elegance of enzyme specificity is also reflected in the work of scientists dedicated to understanding these intricate processes. One such figure is John Kendrew, who, through his pioneering work in protein crystallography, helped elucidate the three-dimensional structures of enzymes. His research illuminated how the precise arrangement of atoms within an enzyme contributes to its specific function. Kendrew once said, "The beauty of life is the beauty of its molecules." This sentiment captures the profound appreciation for the intricate details that define enzyme behavior. Moreover, the specificity of enzymes has inspired numerous advancements in biotechnology. Enzymes are utilized in various applications, from biofuels to food preservation. For instance, proteases are enzymes that break down proteins and are widely used in the food industry to improve digestion and enhance flavor profiles. Their specificity allows for targeted protein breakdown, resulting in products that are not only more flavorful, but also easier to digest. In the realm of medicine, the specificity of enzymes plays a crucial role in drug design. Enzyme inhibitors are often developed to target specific enzymes involved in disease processes. For example, statins are a class of drugs designed to inhibit HMG-CoA reductase, an enzyme critical in cholesterol synthesis. This targeted approach demonstrates how understanding enzyme specificity can lead to effective therapeutic strategies, underscoring the importance of these biological catalysts in our health. As we reflect on the myriad ways in which enzyme specificity influences our lives, we may ask ourselves, how does this knowledge empower us to make informed decisions about our health and the environment? Understanding the role of enzymes can lead to better dietary choices, informed use of medications, and enhanced appreciation for the delicate balance of life. In exploring the intricate relationship between enzymes and metabolic pathways, we find a world where specificity and adaptability coexist. This relationship is not merely a scientific curiosity, but a cornerstone of life itself. Enzymes, with their remarkable ability to tailor their actions to specific substrates while remaining responsive to environmental cues, exemplify the beauty of life's molecular machinery. They remind us that within the complexity of biological systems lies a profound simplicity, the pursuit of balance and efficiency in the dance of life. The dynamic interplay between enzyme specificity and affinity extends beyond mere biochemical interactions, illuminating the profound intricacies of life itself. Each enzyme's ability to recognize its substrate with remarkable precision not only facilitates essential reactions, but also underscores the delicate balance maintained within metabolic pathways. Imagine a finely tuned clock, where each gear must align perfectly for the entire mechanism to function seamlessly. This is the essence of enzymatic specificity. Every enzyme acts as a precise cog, ensuring that life's clock ticks harmoniously. In exploring specificity further, we encounter the concept of enzyme kinetics, which delves into the rates of enzyme-catalyzed reactions. One of the most foundational models is Michaelis-Menten kinetics, which describes how the rate of reaction depends on substrate concentration. Picture a bustling cafe during morning rush hour, where the barista's enzymes serve drinks, products to the customers' substrates. Initially, as more customers arrive, the baristas can keep up, quickly preparing beverages. However, as the line grows longer, the rate at which drinks are served begins to plateau as each barista reaches their capacity. This scenario mirrors how enzymes behave when substrate concentrations increase until they reach a point of saturation. The significance of Michaelis-Menten kinetics lies in its ability to quantify enzyme activity. The Michaelis constant, reflecting the affinity of an enzyme for its substrate, provides insight into how efficiently an enzyme can catalyze a reaction. A low Michaelis constant indicates a high affinity, suggesting that the enzyme can effectively bind to its substrate even at low concentrations. This concept is vital in understanding how enzymes function in varying physiological conditions, adapting their activity to meet the demands of the organism. Consider the role of this adaptability in the context of cellular respiration. In this critical metabolic pathway, enzymes such as phosphofructokinase exhibit a keen sensitivity to the levels of ATP, the energy currency of the cell. When ATP levels are high, phosphofructokinase's activity decreases, effectively slowing down the pathway to prevent an excess of energy. Conversely, when ATP levels drop, the enzyme's activity increases, facilitating the conversion of glucose into energy. This elegant feedback mechanism showcases the adaptability and specificity of enzymes as they respond to the metabolic needs of the cell. The relationship between enzyme specificity and metabolic pathways is further enriched by the concept of allosteric regulation. Allosteric enzymes possess sites separate from their active sites that can bind regulatory molecules, influencing the enzyme's activity. Imagine a conductor guiding an orchestra, where subtle changes in the conductor's movements can shape the performance of the musicians. Similarly, when a regulatory molecule binds to an allosteric site, it alters the enzyme's shape, enhancing or inhibiting its activity. This fine-tuning is crucial for maintaining homeostasis within the cell, allowing for rapid responses to changing environmental conditions. Fascinatingly, the specificity of enzymes also extends to their evolutionary history. Enzymes are not static entities. They evolve in response to changing environments and substrate availability. The concept of enzyme promiscuity emerges, where some enzymes can catalyze multiple reactions and interact with various substrates. While specificity is vital for efficiency, this promiscuity can provide a survival advantage, allowing organisms to adapt to new substrates or environmental challenges. Picture a versatile artisan who can create diverse works, adjusting their skills to meet the demands of different clients. The study of enzyme promiscuity has unveiled remarkable insights into evolutionary biology. For instance, researchers have identified ancestral enzymes with broad substrate specificities that later evolved into more specialized forms. This evolutionary journey highlights the adaptability of enzymes and their ability to refine their functions over time. The interplay between specificity and promiscuity demonstrates a delicate balance, where enzymes must maintain their core functions while also exploring new pathways for survival. As we reflect upon the vast implications of enzyme specificity, we recognize its critical role not only in individual biochemical reactions, but also in the broader context of life's complexity. Enzymes are the architects of metabolic networks, ensuring that each reaction unfolds with precision, contributing to the overall harmony of cellular processes. Their ability to adapt, respond, and evolve serves as a testament to the resilience of life at the molecular level. In contemplating these intricate layers of enzyme specificity and affinity, we may wonder how this knowledge could influence future scientific advancements. Will our understanding of these molecular catalysts pave the way for innovative therapies or sustainable practices? The potential is vast, as the exploration of enzymes continues to unlock new frontiers in medicine, biotechnology, and environmental science. In this journey through the world of enzymes, we discover not only the mechanisms that sustain life, but also the profound interconnectedness of all living systems. Enzymes are not merely catalysts, they are the embodiment of nature's ingenuity, reflecting the elegance and complexity inherent in the web of life. As we delve deeper into this subject, we unveil the remarkable story of enzymes, an ongoing narrative that continues to inspire curiosity and wonder about the molecular dance of existence. Chapter 4: Regulation of Enzyme Activity. Enzymes, while remarkable as biological catalysts, do not operate in isolation. Their activity is finely tuned by various regulatory mechanisms that ensure metabolic pathways function smoothly and efficiently. One of the most fascinating forms of regulation is allosteric regulation. Picture an orchestra where each musician plays their part harmoniously, yet the conductor influences the tempo and dynamics. In the same way, allosteric enzymes possess sites distinct from their active sites, known as allosteric sites. When specific molecules bind to these sites, they induce conformational changes that can either enhance or inhibit the enzyme's activity. Imagine a gentle breeze shifting the direction of a sailboat. This subtle influence can redirect the entire journey. Allosteric regulation is vital for maintaining homeostasis within biological systems. For instance, consider the enzyme phosphofructokinase, a key player in the glycolytic pathway. This enzyme is intricately involved in glucose metabolism, responding to the energy needs of the cell. When ATP concentration is high, indicating ample energy, ATP binds to the allosteric site of phosphofructokinase, causing a shift in its structure that reduces its activity. Conversely, when energy levels are low, ADP binds to the allosteric site, stimulating the enzyme to increase its activity and promote glucose breakdown. This elegant feedback loop ensures that the cell does not waste resources when energy is plentiful, highlighting the grace with which life manages its energy sources. As we journey deeper into the realm of enzyme regulation, we encounter feedback inhibition, another sophisticated mechanism governing metabolic pathways. This process acts as a self-regulating circuit within cellular metabolism, preventing the overproduction of metabolites. Picture a bustling factory where production is carefully monitored. If a certain product reaches surplus levels, the factory must adjust its output to maintain balance. In biochemical terms, when a metabolic pathway produces an end product that accumulates, it often serves as an inhibitor for an earlier enzyme in the pathway. This ensures that resources are conserved and the cellular environment remains in equilibrium. Take the example of the amino acid isoleucine, which is synthesized through a series of enzymatic reactions. Once isoleucine is abundant, it binds to the first enzyme in its biosynthetic pathway, effectively halting further production. This dynamic illustrates a profound principle of biological organization, that systems often contain intrinsic checks and balances to maintain stability. Imagining these regulatory mechanisms invites us to consider the elegance of cellular operation. Why do cells need such intricate systems? The answer lies in the complexity of life itself, where an organism must constantly adapt to changing environments, energy demands, and nutrient availability. The ability to finely tune enzyme activity is a testament to the sophistication of evolutionary processes that have shaped living organisms over billions of years. In addition to allosteric regulation and feedback inhibition, cofactors and coenzymes play crucial roles in the function of enzymes. These molecules are often essential partners that assist enzymes in catalyzing reactions, enhancing their efficiency and capabilities. Visualize a skilled artist who relies on a variety of tools to create a masterpiece. Similarly, enzymes often require cofactors, ions or small molecules, to perform their tasks. These may include metal ions like zinc, magnesium, or iron, which can stabilize enzyme structures or participate directly in catalysis. Coenzymes, a specific subset of cofactors, are organic molecules that bind to enzymes, frequently derived from vitamins. Imagine a well-tuned machine where each component works in unison to achieve a common goal. Coenzymes act as flexible partners, facilitating the transfer of chemical groups between substrates. For instance, the coenzyme NAD+ plays a pivotal role in redox reactions, crucial for energy production. It accepts electrons during the breakdown of glucose in cellular respiration, transforming into NADH in the process. This transformation not only aids in energy extraction, but also highlights the interconnectedness of metabolic pathways, where coenzymes serve as bridges between various reactions. The remarkable synergy between enzymes and their cofactors, or coenzymes, is beautifully illustrated in the case of the enzyme pyruvate dehydrogenase, which plays a vital role in linking glycolysis to the citric acid cycle. This enzyme requires several cofactors, including thiamine, riboflavin, and niacin, each derived from B vitamins. The interplay of these components exemplifies how enzymes are not merely isolated entities, but rather part of a larger network of biochemical interactions that sustain life. As we ponder the role of these essential partners, we might ask, what happens when cofactors are absent or deficient? The consequences can be profound, leading to metabolic disorders or inefficiencies. For example, a deficiency in thiamine can lead to beriberi, a condition that affects energy metabolism. This illustrates the delicate balance that must be maintained for optimal enzyme function and overall health. To further appreciate the importance of cofactors and coenzymes, consider the historical context. The discovery of vitamin B1, or thiamine, marked a turning point in understanding nutrition and its impact on health. It was the work of scientists like Casimir Funk who first identified the necessity of these compounds, linking them to the prevention of diseases. This breakthrough provided a new lens through which to view human health and emphasized the role of dietary components in enzymatic function. In the realm of scientific inquiry, the study of enzyme regulation continues to unfold, revealing layers of complexity and interdependence. Researchers employ advanced techniques to observe these processes in real time, shedding light on how enzymes adapt to their environments and respond to various signals. Imagine peering into a bustling cityscape, where each building represents a different enzyme, working tirelessly to maintain the flow of life. This dynamic landscape is a testament to the continuous evolution of biological systems and their remarkable capacity for adaptation. As we reflect on the intricate mechanisms that regulate enzyme activity, we begin to appreciate the beauty and complexity of life at the molecular level. Each regulatory pathway, each cofactor, and each enzyme plays a vital role in the grand orchestra of biological processes. The delicate balance of activity and inhibition, the intricate dance of molecules, and the interdependent relationships among them form a harmonious symphony, essential for sustaining life as we know it. The intricate dance of enzyme regulation is further enriched by the roles of cofactors and coenzymes, which bind to enzymes and often determine their catalytic capabilities. Imagine a symphony orchestra, where the conductor brings the musicians together, guiding their performance. In this analogy, the enzymes are the musicians, while the cofactors and coenzymes serve as the conductor, orchestrating the necessary cooperation for successful catalytic events. Cofactors can be divided into two main categories: inorganic ions and organic molecules. Inorganic cofactors, like metal ions, play pivotal roles in stabilizing enzyme structures or participating directly in the catalytic process. For instance, zinc ions are critical for the function of numerous enzymes, including carbonic anhydrase, which facilitates the conversion of carbon dioxide and water to bicarbonate and protons. This reaction is central to maintaining acid-base balance in the body, highlighting how the presence of a simple metal ion can have profound implications for physiological processes. On the other hand, coenzymes, which are organic molecules often derived from vitamins, provide a more dynamic role in enzyme catalysis. They can act as carriers of various chemical groups, transferring them between different substrates during metabolic reactions. Imagine a bustling marketplace where goods are exchanged. Coenzymes facilitate the movement of essential molecules, ensuring that the metabolic pathways remain fluid and interconnected. One of the most well-known coenzymes is coenzyme A, which is vital in the metabolism of fatty acids and carbohydrates. It acts as a carrier of acyl groups, effectively linking various metabolic pathways. When fatty acids are broken down for energy, coenzyme binds to these molecules, enabling their entry into the citric acid cycle. This integration illustrates the beauty of biochemical connectivity, where the roles of enzymes, cofactors, and coenzymes intertwine seamlessly. As we explore further, it becomes evident that the absence of these cofactors and coenzymes can lead to significant metabolic disruptions. Consider the case of riboflavin, a vitamin that serves as a precursor for the coenzymes flavin mononucleotide and flavin adenine dinucleotide. These cofactors are essential for a variety of enzymes involved in energy production and cellular respiration. A deficiency in riboflavin can lead to a cascade of effects, resulting in impaired energy metabolism. This underscores the delicate balance that sustains life, where each component, enzymes, cofactors, and coenzymes must be present and functioning harmoniously. The story of enzyme regulation also includes the remarkable phenomenon of enzyme inhibition, which can occur through various mechanisms, including competitive and non-competitive inhibition. In competitive inhibition, an inhibitor molecule competes with the substrate for binding at the enzyme's active site. Visualize a crowded theater where multiple actors vie for the spotlight. The presence of an inhibitor can momentarily obscure the substrate's access to the enzyme, slowing down the reaction. Non-competitive inhibition, however, tells a different story. Here, the inhibitor binds to a site distinct from the active site, altering the enzyme's shape and function without directly blocking substrate access. This mechanism serves as a powerful reminder of the complexity inherent in biological regulation. The elegant balance of enzyme activity does not solely rest on the presence of substrates or the absence of inhibitors. The environment in which enzymes operate also plays a crucial role. Factors such as pH and temperature can significantly influence enzyme function. Imagine a delicate flower thriving in the warm, gentle sunlight. Similarly, enzymes have optimal conditions under which they can perform their best. Deviations from these conditions can lead to decreased activity or even denaturation, where the enzyme loses its functional shape entirely. This intricate relationship with the environment is beautifully illustrated by the enzyme pepsin, which operates optimally in the acidic conditions of the stomach. Pepsin is essential for the breakdown of proteins into smaller peptides, a critical first step in digestion. The acidic pH facilitates the unfolding of protein structures, allowing pepsin to access its substrates effectively. This specificity for environmental conditions exemplifies how enzymes are finely tuned to their roles in the larger context of metabolic pathways. As we continue to unravel the tapestry of enzyme regulation, it becomes increasingly clear that the study of these biological catalysts is not merely an examination of isolated reactions, but rather an exploration of interconnected systems. The elegance of life lies in its complexity, where every piece contributes to the whole. The regulation of enzyme activity reflects a balance of inhibition, activation, environmental factors, and the essential partnerships with cofactors and coenzymes. Imagine a grand performance where each element plays a critical role in creating a masterpiece, a vibrant expression of life's chemistry. This symphony of interactions not only sustains individual cells, but also supports the intricate networks that define living organisms. In this context, we may wonder about the implications of enzyme regulation on health and disease. Disruptions in enzyme activity can result in metabolic disorders, where the delicate balance is thrown into disarray. For instance, phenylketonuria, a genetic condition, arises from a deficiency in the enzyme phenylalanine hydroxylase. This enzyme is crucial for the metabolism of the amino acid phenylalanine. When it is absent or inactive, phenylalanine accumulates to toxic levels, leading to severe neurological consequences. This example serves as a poignant reminder of how integral enzymes are to our well-being and how their regulation is essential for maintaining health. The exploration of enzyme regulation is a journey into the heart of biochemistry, illuminating the pathways that sustain life. Each mechanism of regulation, from allosteric influences to environmental factors, contributes to the remarkable adaptability of living systems. As we reflect on these concepts, we find ourselves at the intersection of science and wonder, where the complexities of life unfold before us, revealing the intricacies of existence in all its forms. Chapter 5: Enzymes in Metabolic Pathways. In the intricate realm of biochemistry, metabolism is often described as the sum of all chemical reactions that occur within a living organism. These reactions are categorized into two primary pathways: catabolic and anabolic pathways. Catabolic pathways break down complex molecules into simpler ones, releasing energy in the process. Picture a vast, ancient tree, its branches heavy with fruit. When the fruit is harvested and consumed, it represents the breakdown of complex carbohydrates into simpler sugars, unlocking energy that fuels our bodies. This energy is vital, propelling us through our daily activities, from the gentle rise of dawn to the quiet lull of night. On the other hand, anabolic pathways construct complex molecules from simpler units, requiring energy input. Imagine a master builder assembling a grand cathedral, each stone carefully placed to create a magnificent structure. The synthesis of proteins from amino acids exemplifies this anabolic process, as our bodies build the very components necessary for growth, repair, and maintenance. These pathways are not isolated. They are interconnected, forming a delicate balance that sustains life. Enzymes play a crucial role in facilitating these metabolic pathways. They act as catalysts, ensuring that reactions proceed smoothly and efficiently. When we think of enzymes, it is important to envision them as skilled artisans, each one expertly crafting its specific product. In catabolic pathways, enzymes break down substrates, liberating energy that can be harnessed for various cellular functions. For example, the enzyme amylase initiates the breakdown of starch into sugars in our mouths, demonstrating how the journey of metabolism begins even before food reaches our stomachs. Consider the Krebs cycle, a pivotal catabolic pathway that occurs within the mitochondria of our cells. This cycle, also known as the citric acid cycle, is a series of enzymatic reactions that transforms acetyl-CoA, derived from carbohydrates, fats, and proteins, into carbon dioxide and water while producing energy-rich molecules such as ATP, NADH, and FADH2. As the cycle turns, each enzyme acts like a cog in a well-oiled machine, facilitating the conversion of energy stored in food into a usable form for the cell. It is remarkable to think that through these enzymatic actions, the very air we breathe becomes a source of energy. One of the most distinguished figures in biochemistry, Hans Krebs, who elucidated the cycle that bears his name, remarked, "The cycle is a series of steps that convert the energy contained in food into the energy of life." His work has illuminated our understanding of how enzymes drive the biochemical reactions that sustain life, revealing a world where energy transformation occurs seamlessly within the confines of our cells. The integration of enzyme actions in cellular metabolism paints a vivid picture of cooperation and harmony. Each enzyme is not merely an isolated entity, but part of a larger network of reactions. Think of a bustling marketplace, where each vendor contributes to the overall vibrancy of the community. In metabolism, enzymes work in concert to ensure that substrates are processed efficiently and effectively. For instance, within the glycolytic pathway, a series of enzymes convert glucose into pyruvate, generating ATP in the process. Each step is meticulously designed, with specific enzymes catalyzing each reaction, ensuring that energy is not wasted and that the cell operates at optimal efficiency. Enzymes are not only essential for breaking down molecules, they also play a vital role in the synthesis of macromolecules. In anabolic pathways, enzymes such as DNA polymerase facilitate the assembly of nucleotides into DNA strands. Imagine a skilled weaver, intertwining threads to create a beautiful fabric. Similarly, enzymes weave together the building blocks of life, constructing the very molecules that carry genetic information and enable the continuity of life itself. The interplay between catabolic and anabolic pathways exemplifies the dynamic nature of metabolism. It is a continuous cycle of building and breaking down, a dance that sustains life. When energy is abundant, anabolic pathways flourish, allowing the organism to grow and thrive. Conversely, during times of scarcity, catabolic pathways take precedence, ensuring that stored energy is mobilized to meet the cell's needs. This adaptability is a testament to the elegance of metabolic regulation, a delicate balance that is finely tuned by the actions of enzymes. As we explore further, consider how enzymes facilitate not just individual reactions, but entire cycles that reflect the ebb and flow of life. The Krebs cycle, for instance, is not merely a sequence of events, but a testament to nature's ability to recycle matter and energy. Each turn of the cycle regenerates important molecules, such as oxaloacetate, ensuring that the process can continue uninterrupted. The efficiency of this cycle is a marvel. It allows living organisms to convert the energy from food into forms that can be utilized for growth, maintenance, and reproduction. The integration of enzyme actions can also be seen in the intricate regulation of metabolic pathways. Enzymes are subject to various factors that modulate their activity, ensuring that biochemical processes are aligned with the needs of the organism. Feedback inhibition, for instance, acts as a regulatory mechanism where the end product of a metabolic pathway inhibits an enzyme involved in its synthesis. This is akin to a conductor signaling an orchestra to lower the volume when the music becomes too loud. By controlling enzyme activity, cells maintain homeostasis, ensuring that resources are not squandered and that metabolic pathways function harmoniously. Another fascinating aspect of enzyme function is their ability to work within specific conditions. Each enzyme has an optimal temperature and pH at which it performs best, much like a delicate flower that blooms under the right conditions. For example, pepsin, an enzyme found in the stomach, thrives in the acidic environment, breaking down proteins into smaller peptides. Conversely, enzymes in the intestines prefer a more neutral pH, where they continue the process of digestion. This specialization highlights the adaptability of enzymes, allowing them to thrive in various environments while fulfilling their roles in metabolism. As we imagine these biochemical pathways, we can visualize the vibrant activity within our cells. Picture the bustling metropolis of enzymes, each one working tirelessly to convert substrates into products. The rhythmic pulse of energy being generated, the harmonious flow of molecules, and the intricate interactions between enzymes and their substrates create a symphony of life. In this world of enzymes, we encounter not just the molecular machinery of life, but also the remarkable individuals who have dedicated their lives to uncovering these secrets. Scientists like Arthur Kornberg, who discovered DNA polymerase, have paved the way for our understanding of how enzymes facilitate the replication of genetic material, enabling life to perpetuate itself across generations. His assertion that the discovery of the enzyme was a major breakthrough encapsulates the significance of enzymes in our understanding of biology. As we continue our exploration of the remarkable world of enzymes and metabolic pathways, let us reflect on how these molecules shape our existence. Each enzymatic action, each metabolic cycle, contributes to the intricate web of life that sustains us. The beauty of this system lies not only in its complexity, but also in its elegance, a reminder of the interconnectedness of all living things. The journey through the landscape of enzymes and their role in metabolism is one that invites us to marvel at the wonders of life itself, revealing the extraordinary processes that allow us to thrive in a world rich with energy and potential. In the ever-evolving landscape of cellular metabolism, enzymes serve as the vital links that harmonize the delicate balance between catabolic and anabolic pathways. The intricacies of their function extend beyond mere catalysis. They embody the essence of communication within the cell. Each enzyme, with its unique attributes and responses to the cellular environment, acts not just as a facilitator, but as a key player in the orchestration of life's processes. Imagine the Krebs cycle not merely as a series of chemical reactions, but as a living narrative, rich with characters and plot twists. Each enzyme in this cycle has a distinct role, akin to actors delivering their lines in a carefully scripted play. For instance, citrate synthase, the first enzyme in the cycle, initiates the transformation of acetyl-CoA and oxaloacetate into citrate, setting the stage for subsequent reactions. This transition is not just a step in a biochemical pathway. It is the opening act of a performance that will ultimately yield energy for the cell. As we picture this cycle, consider how each enzyme's structure is finely tuned to its specific task. Enzymes can undergo conformational changes, adapting their shapes to better fit substrates, much like how a dancer shifts their movements to match the rhythm of music. This flexibility allows enzymes to respond to the fluctuations in cellular conditions, ensuring that metabolic processes can adjust to meet the immediate needs of the organism. In the realm of biochemical cycles, the importance of feedback mechanisms cannot be overstated. They represent the wisdom of evolution, fine-tuning the performance of enzymes based on the cell's requirements. For instance, when energy levels are adequate, the accumulation of certain metabolites can signal enzymes to slow down their activity, preventing unnecessary resource expenditure. This dialogue among enzymes and substrates reflects an elegant regulatory system, where the cell can maintain homeostasis, adjusting its metabolic output in real time. Consider the role of allosteric enzymes once more, as they embody the harmony of regulation. These enzymes possess the remarkable ability to change their activity in response to the binding of effector molecules. This is akin to a painter adding a splash of color to a canvas, transforming a simple image into a vibrant masterpiece. Allosteric regulation allows enzymes to act as sensors, responding to the metabolic state of the cell and ensuring that the pathways remain aligned with the organism's overall needs. The enzymes involved in glycolysis provide another beautiful illustration of this concept. As glucose is transformed into pyruvate, key regulatory enzymes like phosphofructokinase and hexokinase adjust their activity based on the availability of substrates and the energy demands of the cell. It is fascinating to think about how the cell can prioritize energy production when it is abundant while conserving resources during times of scarcity. This adaptability is critical for survival, showcasing the elegant interplay of anabolic and catabolic pathways. Moreover, the integration of enzyme actions extends beyond individual pathways. The interconnectedness of metabolic networks is a testament to the complexity of life itself. For instance, consider how the products of the Krebs cycle feed into various biosynthetic pathways, including amino acid and nucleotide synthesis. The cell's ability to channel intermediates from one cycle to another demonstrates a seamless flow of materials and energy, much like a river that nourishes the surrounding landscape. As we delve deeper into the realm of enzymes, we encounter the groundbreaking work of scientists who have unveiled the intricacies of these catalytic wonders. Consider the contributions of Emil Fischer, who articulated the concept of the lock-and-key model, allowing us to visualize the precise fit between enzymes and substrates. His insights laid the groundwork for our understanding of enzyme specificity and catalysis, emphasizing how nature's design is both practical and remarkably efficient. In addition to Fischer,
the contributions of researchers like DWKTK, HK, 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 HK,
Chapter 6: Enzymes and Human Health. Enzymes play an indispensable role in the complex processes of digestion, where they serve as the key players in breaking down nutrients into forms that our bodies can absorb and utilize. Imagine the journey of food as it enters the mouth, a delightful array of colors and textures, each morsel a potential source of nourishment. As we chew, enzymes like amylase begin their work in the saliva, breaking down carbohydrates into simpler sugars, setting the stage for more intricate biochemical transformations. Once food reaches the stomach, the environment shifts dramatically. Here, gastric juices, rich in hydrochloric acid and pepsin, work together to further break down proteins into peptides. Picture the stomach as a cauldron, simmering with activity, where enzymes dance in a symphony of chemical reactions. This process continues as food moves into the small intestine, where pancreatic enzymes such as lipase, protease, and amylase join the mix. Together, they dismantle fats, proteins, and carbohydrates into their constituent parts: fatty acids, amino acids, and simple sugars. This breakdown is critical, as it transforms complex food into the essential building blocks that fuel our cells, tissues, and organs. Yet, what happens when this finely tuned machinery falters? Enzyme deficiencies can lead to a range of disorders that affect digestion and overall health. Consider lactose intolerance, a condition stemming from the deficiency of lactase, the enzyme responsible for breaking down lactose, the sugar found in milk. Individuals with this deficiency may experience discomfort and digestive distress after consuming dairy products. This condition illustrates the crucial balance enzymes maintain in our bodies. Without the right enzymes, even the most wholesome foods can become sources of discomfort rather than nourishment. Another example is phenylketonuria, a genetic disorder caused by the inability to produce the enzyme phenylalanine hydroxylase. This enzyme is essential for metabolizing phenylalanine, an amino acid found in many protein-rich foods. When untreated, this deficiency can lead to serious neurological issues. Such conditions remind us of the delicate interplay between enzymes and health, where the absence of a single enzyme can have profound implications. The role of enzymes extends beyond digestion. They are also pivotal in the realm of pharmacology and drug design. Imagine a world where medications are tailored to interact precisely with specific enzymes in the body. This precision can lead to more effective treatments with fewer side effects. Enzymes are often targets in drug design because they are involved in many biological pathways. By understanding the structure and function of these enzymes, scientists can develop inhibitors that block their activity, offering therapeutic benefits. Take, for example, the development of statins, a class of drugs that lower cholesterol levels. These medications work by inhibiting HMG-CoA reductase, an enzyme crucial for cholesterol synthesis in the liver. By blocking this enzyme, statins effectively reduce the amount of cholesterol produced, helping to prevent cardiovascular diseases. Imagine the impact this discovery has had on public health, saving countless lives and improving the quality of life for many. Moreover, advances in biotechnology have opened new avenues for enzyme applications in medicine. Enzyme replacement therapy, for instance, uses enzymes to treat disorders that arise from enzyme deficiencies. Conditions like Gaucher's disease, where the body lacks the enzyme glucose reversidase, can be managed through the administration of synthetic or recombinant enzymes. This treatment not only alleviates symptoms, but also enhances the quality of life for affected individuals, illustrating the profound potential of enzymes beyond their natural roles. As we explore the multifaceted nature of enzymes, we can appreciate how they serve as both facilitators of essential processes and as targets for innovative therapies. They remind us of the intricate balance within our bodies, where a slight disruption can lead to significant health challenges. Think of enzymes as the unsung heroes of our biological systems, tirelessly working behind the scenes to ensure that life continues to flourish. In reflecting upon the interconnectedness of enzymes and human health, we must ask ourselves, how can we support these vital molecules? Nutrition plays a crucial role in this equation. A balanced diet, rich in whole foods, can provide the necessary substrates that enzymes require to function optimally. Foods rich in vitamins and minerals, such as leafy greens, nuts, and seeds, contain cofactors that enhance enzyme activity. Imagine vibrant fruits and vegetables, each a colorful representation of the nutrients that support our health. Additionally, understanding the effects of lifestyle choices on enzyme function can empower us to make informed decisions. Regular physical activity, for instance, can enhance metabolic pathways and promote the efficient functioning of enzymes involved in energy production. The benefits of exercise extend beyond physical fitness. They create a harmonious environment where enzymes can thrive, contributing to overall well-being. As we consider the future of enzyme research, the possibilities seem boundless. Scientists are continually exploring the vast potential of enzymes in various fields, from sustainable agriculture to biofuels. Enzymes are being harnessed to create more efficient processes that reduce waste and environmental impact. Imagine a world where enzymes play a central role in producing clean energy, transforming waste into valuable resources, and promoting ecological balance. In the context of human health, the exploration of enzymes continues to deepen our understanding of the mechanisms that sustain life. The more we learn about these remarkable molecules, the clearer it becomes that they are at the heart of our biological processes. They not only facilitate the digestion of our food, but also hold the key to innovative medical treatments and advancements in healthcare. As we embark on this journey through the world of enzymes, let us remain open to the wonders of scientific discovery. Each new revelation about enzymes offers a glimpse into the intricate workings of life itself, encouraging us to appreciate the delicate balance that sustains our health and well-being. Imagine the stories yet to be uncovered, the connections yet to be made, as we explore the vast terrain of enzymes and their impact on our lives. Each finding is a step toward greater understanding, illuminating the path for future generations to follow. The harmony of enzymes within our bodies serves as a reminder of the complexity of life. They are the catalysts of health, weaving through the fabric of our existence, ensuring that each process unfolds with grace and precision. As we continue to investigate their myriad roles, we stand to gain not only insights into our own biology, but also the potential to enhance the health of our communities and our planet. Enzymes are not only vital for digestion and metabolic processes, they also hold significant promise in the field of pharmacology and drug design. This aspect of enzymes is particularly fascinating, as it allows us to explore how these biological catalysts can be harnessed to develop innovative treatments for various health conditions. Imagine a world where medications are crafted to interact precisely with specific enzymes, enhancing their efficacy while minimizing side effects. This vision is increasingly becoming a reality. Consider the role of enzymes in the development of targeted therapies. Researchers have discovered that many diseases, including cancer, arise from the dysregulation of enzymatic activity. By identifying the specific enzymes involved in disease pathways, scientists can design drugs that inhibit or enhance their function. This targeted approach is akin to using a fine-tipped brush to paint intricate details on a canvas, ensuring that treatment is both precise and effective. One striking example is the development of protease inhibitors used in the treatment of HIV. The HIV virus relies on proteases to process its proteins and replicate within the host. By designing drugs that specifically inhibit these viral proteases, researchers have created effective treatments that disrupt the virus's life cycle. This innovative strategy has transformed the management of HIV, allowing individuals to live longer, healthier lives. Another compelling aspect of enzyme research lies in the use of enzymes in personalized medicine. Each individual possesses a unique genetic makeup, which can influence how their body processes medications. By understanding the specific enzymes involved in drug metabolism, healthcare providers can tailor treatments to suit individual needs. Imagine a scenario where your medication is adjusted based on how your body metabolizes the drug, optimizing its effectiveness while reducing the risk of adverse effects. This level of personalization represents a significant leap forward in healthcare. Enzymes also play an essential role in the field of biotechnology. Scientists are increasingly utilizing enzymes in the development of novel therapies, such as gene editing technologies. The CRISPR-Cas9 system, which has garnered immense attention for its potential to edit genes with precision, relies on enzymes that can cut DNA at specific locations. This powerful tool opens up possibilities for treating genetic disorders, allowing researchers to correct mutations that lead to disease. Imagine the hope that this technology brings to individuals with inherited conditions, as it offers the prospect of curing previously untreatable ailments. Furthermore, the potential of enzyme-based therapies extends to the management of chronic conditions. For instance, enzyme replacement therapy is a revolutionary approach for individuals with enzyme deficiencies. Conditions such as Fabry disease, where the body lacks the enzyme alpha-galactosidase, can be effectively managed through the administration of synthetic enzymes. By replacing the missing enzyme, patients experience a significant improvement in their symptoms and quality of life. This therapeutic strategy underscores the importance of enzymes not only in digestion, but also in the maintenance of overall health. As we delve deeper into the role of enzymes in health, we must consider the impact of lifestyle choices on their function. Nutrition, as previously mentioned, plays a crucial role in supporting enzymatic activity. A diet rich in diverse, whole foods provides the necessary cofactors and substrates that enzymes require to perform their functions optimally. Think of vibrant fruits and vegetables as the fuel that empowers enzymes to work efficiently, creating a vibrant symphony of biological processes. Moreover, staying hydrated is equally important. Water is essential for many enzymatic reactions, as it often serves as a medium for the biochemical interactions that occur within our bodies. Imagine the fluidity of water, facilitating the movement of molecules, allowing enzymes to access their substrates effortlessly. Adequate hydration can enhance enzyme activity, promoting optimal digestion and metabolic function. Physical activity, too, exerts a profound influence on enzyme function. Regular exercise stimulates various metabolic pathways and enhances the activity of enzymes involved in energy production. Picture the invigorating flow of movement, where each step echoes the harmonious interplay of enzymes working to convert stored energy into fuel. This synergy between exercise and enzyme activity not only boosts physical health, but also uplifts mental well-being. As we consider the future of enzyme research, we are reminded of the immense potential that lies ahead. The exploration of enzymes in medicine, from drug design to gene editing, continues to expand our understanding of their roles in health and disease. Each discovery holds the promise of innovative treatments that could revolutionize how we approach healthcare, emphasizing the importance of a personalized approach to medicine. Imagine a future where chronic illnesses can be managed more effectively, where therapies are tailored to the unique enzymatic profiles of individuals, and where the mysteries of genetic disorders can be unraveled with newfound clarity. The landscape of health and medicine is evolving, driven by the intricate dance of enzymes that underpin our biological processes. In this journey, scientists are not only unraveling the mysteries of enzymes, but also exploring their ecological roles. Enzymes are being utilized in sustainable practices, offering solutions to environmental challenges. Consider the application of enzymes in bioremediation, where they are employed to break down pollutants and restore ecosystems. This harmonious relationship between enzymes and the environment underscores their versatility and significance beyond human health. As we ponder the future of enzymes, let us remain open to the possibilities that lie ahead. Each breakthrough in enzyme research offers a glimpse into the intricate workings of life, reminding us of the interconnectedness of all living systems. Whether through advancements in medicine or environmental sustainability, enzymes continue to shape our understanding of the world and open doors to innovative solutions. The journey of discovery is ongoing, and as we explore the vast terrain of enzymes, we invite curiosity and wonder into our lives. Each step forward brings us closer to a deeper appreciation of these remarkable molecules, illuminating the profound impact they have on our health, our environment, and the very essence of life itself. Chapter 7: Industrial Applications of Enzymes. In the vibrant world of food processing, enzymes serve as indispensable tools, transforming raw ingredients into the delightful meals we enjoy. Imagine walking through a bustling market, where the aroma of freshly baked bread wafts through the air, inviting you to savor its warmth. This transformation, from simple grains to the complex flavors of bread, is largely facilitated by enzymes in fermentation. Fermentation is a process that relies on enzymes to convert sugars into alcohol or acids, producing a spectrum of flavors and textures. One of the most well-known examples is the fermentation of dough, where yeast, a microorganism rich in enzymes, converts sugars into carbon dioxide and alcohol. Picture the yeast as tiny artisans, each one working tirelessly, producing bubbles of gas that cause the dough to rise, creating a light and airy loaf. As the bread bakes, the heat deactivates the yeast, locking in the complex flavors developed during this magical transformation. But enzymes in food processing extend beyond bread. In the world of dairy, enzymes like rennet play a crucial role in cheese production. Rennet is a complex of enzymes that curdles milk, separating it into solid curds and liquid whey. Imagine the gentle curdling of milk, like a soft embrace, transforming a liquid into a solid form rich in flavor and nutrients. The art of cheese-making exemplifies how enzymes can create diverse textures and tastes, from creamy brie to sharp cheddar, each with its own unique story. Enzymes also find their place in the production of beverages like wine and beer, where they break down complex sugars to release alcohol and contribute to the intricate flavor profiles of these drinks. The process of winemaking, for instance, is a delicate dance of enzymes, as they work to convert the sugars in grapes into alcohol, while also influencing the aroma and taste. Picture a vineyard at dusk, where the sweet scent of fermenting grapes fills the air, the enzymes quietly orchestrating a symphony of chemical reactions, crafting each bottle of wine into a unique experience. As we explore the realm of biotechnology, enzymes unveil their potential in the creation of biofuels and biosensors. Biofuels, derived from organic materials, rely on enzymes to convert biomass into usable energy. Imagine the vast fields of corn or sugar cane, their cells rich with energy, awaiting the catalytic touch of enzymes. These enzymes break down complex carbohydrates into simple sugars, which can then be fermented into ethanol. This process not only provides a renewable energy source, but also reduces our reliance on fossil fuels, painting a picture of a more sustainable future. In the realm of biosensors, enzymes act as sensitive detectors, responding to specific substances with remarkable precision. Consider the way an enzyme can bind to a target molecule, triggering a measurable response. This phenomenon is harnessed in medical diagnostics, where enzymes can detect glucose levels in blood samples, offering crucial insights for individuals managing diabetes. Visualize a tiny sensor, delicately attuned to the presence of glucose, providing real-time feedback that can shape health decisions. Such innovations showcase the versatility of enzymes, transforming them from mere catalysts into essential tools for monitoring health and well-being. Moving beyond the realm of industry, enzymes play a vital role in environmental applications, particularly in bioremediation. This process utilizes enzymes to break down pollutants, restoring ecosystems to their natural state. Imagine a polluted river, its waters murky and lifeless, yearning for renewal. Enzymes can help cleanse such environments by breaking down toxic substances into harmless byproducts. For instance, certain bacteria produce enzymes capable of degrading oil spills, converting the harmful hydrocarbons into benign compounds. Picture tiny organisms working tirelessly, each enzyme a beacon of hope, transforming devastation into restoration. The power of enzymes in bioremediation extends to heavy metal detoxification as well. Certain fungi and bacteria produce enzymes that can bind to heavy metals, facilitating their removal from contaminated environments. This process is akin to nature's own cleanup crew, diligently working to restore balance and health to ecosystems. As we contemplate the potential of enzymes, we are reminded of the words of a renowned environmental scientist who said, "Nature knows no waste. It only knows how to recycle." In this interconnected web of life, the role of enzymes is profound. They not only enhance our food and fuel, but also pave the way for environmental recovery. As we reflect on the contributions of enzymes in industry, we see how they bridge the gap between nature and technology, creating a harmonious balance. These remarkable molecules epitomize the ingenuity of life, embodying the delicate balance of function and purpose. As we journey through the myriad applications of enzymes, we uncover a world where science and nature intertwine, each enzyme a testament to the beauty of biochemical artistry. Imagine the endless possibilities that lie ahead as we continue to explore and harness the power of these biological catalysts in various fields. The future is bright, and with every innovation, we embrace the potential of enzymes to transform our world in ways we have yet to fully comprehend. In the vast arena of biotechnology, the influence of enzymes extends even further, illuminating pathways toward sustainable energy solutions. Among these innovations, biofuels emerge as a prominent application, embodying the potential to transform organic materials into renewable energy sources. Imagine fields of corn swaying gently in the breeze, each stalk brimming with energy waiting to be unleashed. Enzymes play a pivotal role in this process by breaking down the complex carbohydrates found in plant biomass into simpler sugars. These sugars can then be fermented into ethanol, a biofuel that serves as a cleaner alternative to fossil fuels. The journey of converting biomass into biofuel showcases the synergy between nature and technology. Enzymatic hydrolysis, a key step in this conversion, involves the action of cellulases, enzymes that break down cellulose, a major component of plant cell walls. Picture these enzymes as skilled artisans, meticulously dismantling the fibrous structure of plants, freeing the sugars that lie within. This transformation not only holds promise for energy production, but also represents a shift towards more environmentally friendly practices. As we consider the potential of biofuels, we are reminded of the words of a visionary in this field who stated, "The future of energy is not just renewable, it's also biological." In addition to biofuels, enzymes have carved out a vital role in the development of biosensors, devices that detect specific substances with remarkable accuracy. These sensors rely on enzymes to catalyze reactions that produce measurable signals, making them invaluable in medical diagnostics and environmental monitoring. Imagine a world where a small device can provide real-time data about one's health or the quality of the air we breathe. Enzymes enable such advancements by binding to target molecules and triggering detectable changes. Take, for example, glucose biosensors, crucial for individuals managing diabetes. These sensors utilize glucose oxidase, an enzyme that catalyzes the oxidation of glucose, producing hydrogen peroxide as a byproduct. This reaction generates an electrical signal proportional to the glucose concentration, offering insight into blood sugar levels. Picture a tiny, unobtrusive device that allows individuals to monitor their health effortlessly, empowering them to make informed decisions about their lifestyle and well-being. The integration of enzymes into biosensor technology exemplifies how science bridges the gap between complexity and clarity, enhancing our ability to navigate health challenges. As we shift focus to environmental applications, the significance of enzymes in bioremediation becomes increasingly evident. Enzymes act as nature's cleanup crew, breaking down pollutants and restoring ecosystems to their natural state. Imagine a river once choked with contaminants, its waters murky and lifeless. Enzymes possess the remarkable ability to metabolize harmful substances, converting them into harmless byproducts and rejuvenating the environment. Consider the impact of oil spills, devastating events that disrupt marine ecosystems. Certain bacteria and fungi produce enzymes capable of degrading hydrocarbons, the primary components of oil. These enzymes catalyze the breakdown of complex oil molecules into simpler, less harmful compounds, facilitating the natural recovery of affected areas. Visualize tiny organisms tirelessly working together, each enzyme a vital player in the restoration process, highlighting the resilience and adaptability of nature. Beyond oil spills, enzymes also contribute to the detoxification of heavy metals from contaminated soils and waters. Some microorganisms have evolved to produce enzymes that can bind to heavy metals, sequestering them and preventing their toxic effects on living organisms. This process is akin to a biological filter, cleansing the environment of harmful substances. Think of the potential for restoring habitats that have suffered from industrial pollution, where enzymes serve as the key agents of renewal. Reflecting on these environmental applications invites us to ponder the intricate relationship between human activity and the natural world. As we create technologies that harness the power of enzymes, we are reminded of our responsibility to protect and preserve the ecosystems that sustain us. The words of an environmental advocate resonate here. We do not inherit the earth from our ancestors, we borrow it from our children. In this context, enzymes not only enhance industrial processes, but also offer solutions to some of the most pressing challenges we face today. Their ability to facilitate complex reactions in a sustainable manner underscores the importance of integrating scientific advancements with environmental stewardship. By embracing the potential of enzymes, we open the door to a future where innovation and nature coexist harmoniously, paving the way for a healthier planet. As we delve deeper into the multifaceted applications of enzymes, it becomes clear that these remarkable molecules are not merely tools of industry. They are vital players in the symphony of life. From the food we consume to the energy we harness and the environments we strive to protect, enzymes serve as the unseen catalysts that drive progress and sustainability. Each application tells a story of connection between science and nature, between innovation and responsibility. With every new discovery, we inch closer to realizing the full potential of enzymes, revealing their ability to shape our world in ways we have yet to fully comprehend. As we continue this exploration, we are reminded that the future, guided by the wisdom of nature, holds boundless possibilities for the applications of these extraordinary biological catalysts. As we turn our attention to the environmental applications of enzymes, we begin to uncover their remarkable potential in bioremediation. This process, where enzymes act as nature's cleanup agents, offers a powerful means to restore balance in ecosystems that have been disrupted by human activity. Imagine a landscape once vibrant, now marred by pollution, its natural beauty dulled by the weight of contaminants. Enzymes step in as facilitators of recovery, breaking down harmful substances into benign components, thereby reviving the health of our planet. Consider the scenario of heavy metal pollution, a pressing issue in many industrial regions. Enzymes produced by certain fungi and bacteria exhibit a unique ability to bind to heavy metals, effectively sequestering these toxic elements. Picture a microscopic world where these organisms thrive, their enzymes acting like sponges, absorbing harmful substances and preventing them from leaching into surrounding soils and waterways. This natural detoxification process is not only efficient, but also elegant, embodying the intricate interplay between life forms and their environments. The story of bioremediation is enriched by the contributions of scientists who have dedicated their lives to understanding and harnessing the power of enzymes. One such figure is the renowned microbiologist who famously stated, "Nature is the best chemist." This perspective underscores the idea that the solutions to our most significant environmental challenges often lie within the very ecosystems we seek to protect. By studying how organisms naturally degrade pollutants, we can develop innovative strategies to address contamination, ensuring a healthier future for our planet. As we envision the applications of enzymes in bioremediation, we must also consider the broader implications of these discoveries. The ability of enzymes to facilitate the breakdown of complex pollutants not only aids in restoring ecosystems, but also presents opportunities for sustainable industrial practices. Imagine industries adopting enzyme-based bioremediation strategies as a standard practice, reducing their environmental footprint while promoting the health of local ecosystems. This shift toward sustainability is not just an ideal. It is a necessary evolution in our approach to industry and environmental stewardship. In addition to their role in pollution cleanup, enzymes serve as vital components in developing eco-friendly processes across various sectors. For example, The textiles industry has historically been associated with significant environmental degradation due to the use of harsh chemicals in dyeing and finishing processes. However, enzymes can offer a more sustainable alternative by facilitating the breakdown of dyes and impurities, resulting in cleaner production methods. Imagine a factory floor where enzymes replace toxic chemicals, creating textiles that are not only beautiful, but also bear the mark of environmental consciousness. The intersection of enzyme technology and environmental health opens up a realm of possibilities. By embracing the wisdom of nature and integrating enzymatic processes into our industries, we can foster a more harmonious relationship with the environment. This journey is not merely about mitigating damage. It is about reimagining our approach to production, consumption, and the stewardship of our natural resources. As we reflect on the multifaceted roles of enzymes in industrial applications, it becomes clear that they are more than just tools for efficiency and productivity. They embody the essence of collaboration between science and nature, revealing pathways toward sustainability that were once thought to be distant dreams. The future of enzyme technology holds immense promise, inviting us to explore and innovate in ways that honor the delicate balance of our ecosystems. Through the lens of enzymes, we gain insights into the intricate connections that bind us to the world around us. Their ability to catalyze transformative reactions resonates not only within laboratories and factories, but also in the very fabric of our lives. Enzymes, with their remarkable adaptability and specificity, remind us of the elegance inherent in nature's design, inspiring us to pursue a future where innovation and environmental harmony go hand in hand. Chapter 8: Enzymes in Research and Biotechnology. Enzymes, those remarkable molecules that serve as biological catalysts, are not only crucial for the processes of life, but also hold tremendous potential in the realms of research and biotechnology. Understanding how to isolate and characterize these enzymes opens a world of possibilities, each step revealing the intricacies of their behavior and function. Imagine the journey of an enzyme from its natural habitat to the laboratory. The first step in this journey is isolation, which allows scientists to separate enzymes from the complex mixtures found in biological systems. This process often begins with the extraction of enzymes from tissues, cells, or microorganisms. Techniques vary widely, yet each is designed to preserve the delicate nature of the enzyme while removing unwanted materials. Methods such as centrifugation spin the mixture at high speeds, forcing heavier cellular debris to the bottom of the container, while lighter proteins remain in the solution. Visualize this as a calm lake, where the still waters reflect the sky, and beneath the surface, the sediment settles gently to the bottom, leaving clarity above. Following extraction, purification techniques are employed, including chromatography, which can be likened to a carefully planned journey through a maze. Imagine the enzyme navigating this labyrinth, with each turn designed to separate it from other molecules based on specific characteristics like size or charge. This meticulous process allows researchers to obtain enzymes in a form that is both concentrated and functional, paving the way for further study. Characterization of enzymes is equally vital, as it involves determining their properties and behaviors. Scientists employ various assays to assess enzyme activity, substrate specificity, and optimal conditions for activity. Picture a scientist observing the enzyme as it interacts with its substrate, noting the precise moment of transformation. This stage reveals how enzymes can be influenced by factors such as temperature and pH, which serve as the environmental backdrop for their performance. Each enzyme has an optimal temperature range, much like an artist who thrives in their own unique setting, producing their best work under specific conditions. In recent years, the genetic engineering of enzymes has emerged as a powerful tool in biotechnology. By altering the genetic code that encodes an enzyme, researchers can enhance its performance, stability, and specificity. Imagine a sculptor chiseling away at a block of marble, refining their creation until it embodies beauty and precision. Techniques such as site-directed mutagenesis allow scientists to make specific changes to the enzyme's amino acid sequence, resulting in a modified enzyme that performs better under industrial conditions. This innovative approach has led to the development of enzymes that can withstand extreme temperatures or harsh chemical environments, making them invaluable for various applications, from biofuels to pharmaceuticals. One of the most inspiring figures in the field of enzyme engineering is Frances Arnold, who was awarded the Nobel Prize for her pioneering work on directed evolution. Arnold's research underscores the potential of mimicking natural selection to create enzymes with desired characteristics. She once stated, "I just want to make the world a better place," a sentiment that resonates deeply within the scientific community. Through her work, scientists can envision a future where enzymes are tailored to meet the specific needs of society, contributing to sustainable practices and innovative solutions. As we look toward the future of enzyme research, the field of synthetic biology beckons with promise and intrigue. This interdisciplinary domain combines biology, engineering, and computer science, aiming to design and construct new biological parts, devices, and systems. Enzymes are at the heart of this exploration, serving as the building blocks for synthetic pathways that can produce valuable compounds. Imagine a world where enzymes are engineered to convert waste into energy, or to synthesize life-saving medications in a more efficient manner. The concept of synthetic biology allows for the creation of entirely new metabolic pathways, enabling organisms to produce substances that do not naturally occur in nature. Envision a tiny factory, an engineered microorganism, that has been programmed to produce biofuels, pharmaceuticals, or even biodegradable plastics. This visionary approach could revolutionize industries, reducing reliance on fossil fuels while promoting sustainable alternatives. Moreover, the integration of computational tools in enzyme research has opened new avenues for discovery. Advanced algorithms can predict enzyme behaviors and interactions, enabling scientists to design enzymes with unprecedented precision. Picture a vast library of information, where each molecule is cataloged, and researchers can sift through data to find enzymes that fit specific criteria. This fusion of biology and technology fosters a deeper understanding of enzyme dynamics, propelling research into uncharted territories. As scientists continue to explore the depths of enzyme capabilities, they remain acutely aware of their responsibility to harness these powerful tools ethically. The potential for misuse exists, and with it comes the need for a thoughtful approach to research and development. Consider the implications of creating enzymes that can break down plastics or those that can be used in gene editing. The need for a framework that guides this research is paramount, ensuring that the benefits of enzyme technology are realized while minimizing risks to our environment and society. In the intricate landscape of research and biotechnology, enzymes stand as beacons of hope and progress. They embody the essence of innovation, merging the wisdom of nature with the creativity of human endeavor. Each discovery leads to new possibilities, each engineered enzyme a testament to our ability to influence the building blocks of life. As we reflect on the journey of enzymes through isolation, characterization, and genetic engineering, we are reminded of their profound impact on our world. What lies ahead in enzyme research is not just a continuation of this exploration, but a leap into a future where biology and technology converge to address some of our most pressing challenges. The story of enzymes is ongoing, rich with potential and promise, waiting to unfold in remarkable ways. The techniques for enzyme isolation and characterization not only unveil the secrets of these biological catalysts, but also set the stage for further innovations in biotechnology. Imagine, if you will, the meticulous work that goes into each experiment, a dance of precision and patience, where scientists employ various methods to reveal the unique properties of enzymes. One such technique is electrophoresis, which allows researchers to separate enzymes based on their size and charge. Visualize this process as a river, where enzymes flow along different currents, some gliding swiftly while others are held back by the rocky bed below. The result is a clear separation, illuminating the diversity of enzymes within a sample. When scientists analyze the bands formed during this process, they gain insight into the enzyme's molecular weight and purity, essential information for the next steps in research. Additionally, enzyme kinetics is a cornerstone of characterization, providing a glimpse into how enzymes interact with their substrates. Picture a race course where different substrates compete for the attention of their respective enzymes. By measuring reaction rates under various conditions, researchers can determine important parameters like the maximum reaction velocity and the Michaelis constant, shedding light on the efficiency of enzyme activity. This quantitative analysis not only helps in understanding enzyme behavior, but also informs the engineering of enzymes for specific applications. Genetic engineering has become a beacon of hope in enhancing enzyme performance, providing tools to modify enzymes at the molecular level. One of the most revolutionary techniques in this domain is CRISPR technology, a method that allows for precise editing of genetic material. Imagine a skilled artist with a brush, able to alter a painting with a few deft strokes. In a similar way, scientists can target specific genes that encode for enzymes, introducing changes that improve their properties. This has led to the creation of enzymes with higher catalytic efficiency or altered substrate specificity, opening doors to applications that were once thought to be unattainable. The story of Jennifer Doudna and Emmanuel Charpentier, pioneers of the CRISPR-Cas9 system, exemplifies the remarkable potential of genetic engineering. They once noted, "The beauty of the CRISPR system is that it can be adapted for a wide variety of uses," highlighting its versatility not just in biology, but in medicine, agriculture, and beyond. This adaptability extends to enzyme engineering, where the ability to tailor enzymes for specific industrial processes is transforming fields such as biomanufacturing and environmental remediation. As we contemplate the future of enzyme research, the burgeoning field of synthetic biology stands at the forefront, offering a canvas on which scientists can paint their visions of what is possible. Enzymes are increasingly being recognized as essential components in the design of synthetic pathways, capable of producing complex molecules that can address global challenges. Imagine a world where engineered microorganisms could convert agricultural waste into biofuels, or where enzymes are designed to selectively degrade pollutants in our water systems. This vision is not merely a dream. It is unfolding in laboratories around the world. The concept of enzyme cascades is particularly fascinating in synthetic biology. Picture a relay race, where each runner passes the baton to the next, facilitating a series of reactions that culminate in the production of a desired product. By engineering multiple enzymes to work in concert, researchers can create efficient pathways that enhance overall yields and reduce byproducts. This approach not only maximizes resource utilization, but also minimizes the environmental impact of chemical processes, aligning with the principles of sustainability. Furthermore, the integration of artificial intelligence into enzyme research is revolutionizing the discovery process. Imagine a vast ocean of data, where each piece of information contributes to a greater understanding of enzyme interactions and functions. AI algorithms can analyze this data, identifying patterns and predicting which modifications will lead to improved enzyme performance. This fusion of computational power with biological research holds the promise of accelerating the pace of discovery, enabling scientists to explore uncharted territories in enzyme engineering. As we delve into the ethical dimensions of enzyme research in synthetic biology, it becomes ever more crucial to navigate the challenges that accompany these advancements. The potential for creating new organisms or altering existing ones raises questions about ecological balance and safety. How do we ensure that the benefits of these technologies are realized without compromising the delicate web of life? This inquiry invites a collective dialogue among scientists, ethicists, and the public, fostering transparency and responsibility in research practices. In this evolving landscape, enzymes wield the power to transform not just industries, but also the very fabric of our society. They represent the intersection of nature and technology, where each discovery brings us closer to sustainable solutions for the challenges we face. As we continue to explore the multifaceted roles of enzymes in research and biotechnology, it is clear that their story is one of endless potential, waiting patiently to unfold in the chapters yet to be written. The journey of enzymes, from their isolation and characterization to their engineering and application, serves as a testament to human ingenuity and our desire to harness nature's wisdom. What further innovations await us? What new horizons will be illuminated by the brilliance of enzymes as we continue to push the boundaries of science? The answers lie in the uncharted depths of research, inviting us to dream, discover, and innovate. Chapter 9: The Impact of Temperature and pH on Enzyme Function. As we explore the delicate balance of temperature and pH in relation to enzyme function, we step into a world where the very essence of life's chemistry is influenced by these two fundamental factors. Enzymes, as we have come to understand, are remarkable catalysts that facilitate biochemical reactions. Yet, their efficiency and functionality are not static. They are profoundly affected by environmental conditions. Imagine a serene pond, where the temperature of the water creates ripples across its surface. In a similar manner, temperature impacts enzyme kinetics, the rate at which enzymes catalyze reactions. Each enzyme has a specific temperature range in which it operates optimally, akin to a musician playing a piece of music that resonates best at a certain tempo. When the temperature rises, the activity of most enzymes increases, leading to a quicker reaction rate. This increase occurs because higher temperatures provide more kinetic energy to the molecules involved, allowing them to collide more frequently and with greater force. However, this relationship is not linear. Picture a gentle slope that rises to a peak where the enzyme operates at its best, before descending into a valley of denaturation. At elevated temperatures beyond the optimal range, enzymes begin to lose their structural integrity. The delicate bonds that hold their three-dimensional shapes together start to break apart, leading to a loss of function. This phenomenon can be visualized as a beautiful sculpture melting under the sun's heat, losing its form and purpose. one of the notable figures in biochemistry, Frederick Sanger, emphasized the importance of temperature in enzyme research, stating that the activity of the enzyme is determined by the temperature at which it is measured. His work laid the foundation for understanding how enzymes behave in varying thermal environments. Let us also consider pH, the measure of acidity or alkalinity in a solution. Enzymes possess specific pH ranges that dictate their activity, just as a plant thrives in particular soil conditions. For many enzymes, a slight deviation from their optimal pH can lead to significant reductions in activity. Imagine a delicate flower wilting when exposed to harsh conditions, unable to flourish as it should. Each enzyme has evolved to perform its function in a specific pH environment. Pepsin, for instance, is an enzyme that works effectively in the acidic environment of the stomach, breaking down proteins into smaller peptides. Picture the stomach as a culinary workshop where pepsin diligently cuts proteins, preparing them for the next stage of digestion. Conversely, enzymes like trypsin operate best in the more alkaline conditions of the small intestine, where the environment becomes conducive to further digestion. This sensitivity to pH is not merely a quirk, it reflects the intricate dance of life at a molecular level. Enzymes are composed of amino acids, and the charges on these amino acids can change depending on the pH of their surroundings. This charge alteration can affect the enzyme's shape and, ultimately, its ability to bind substrates effectively. Rhetorically speaking, how can we harness this knowledge? The answer lies in application. In the industrial realm, the understanding of temperature and pH is pivotal in optimizing enzyme function for various processes. In the brewing industry, for example, brewers carefully control the temperature during fermentation to ensure that yeast enzymes convert sugars into alcohol efficiently. Imagine the satisfaction of a perfectly brewed beer, where every sip is a testament to the precise manipulation of temperature. Similarly, in the world of pharmaceuticals, enzymes are employed to synthesize complex molecules. By adjusting the pH to suit specific enzymes, chemists can enhance reaction rates and yields, leading to more effective and efficient drug production. Visualize a bustling laboratory, scientists focused intently on their work, their efforts guided by the principles of temperature and pH that govern enzyme activity. In the field of food production, enzymes play a crucial role in enhancing flavors and textures. Consider the process of cheese making. The pH of milk is carefully adjusted to promote the activity of rennet, an enzyme that curdles the milk, separating it into curds and whey. This transformation is not just a change in state, but rather a delicate interplay governed by conditions that must be meticulously controlled. As we reflect on the profound impact of temperature and pH on enzyme function, it becomes evident that these factors are not just mere numbers on a scale. They are the unseen forces that shape life's biochemical landscape. They dictate how enzymes perform their magic, allowing nature's processes to unfold with grace and precision. Throughout history, scientists have endeavored to unlock the secrets of enzymes, peering into the molecular world and discovering how these catalysts respond to their environments. The work of early biochemists paved the way for our current understanding, allowing us to manipulate these processes in ways that benefit society. In our exploration, we are reminded of the interconnectedness of life. Just as enzymes rely on specific conditions to function optimally, we too thrive when our environments are balanced. Whether it is the warmth of the sun on our skin or the gentle breeze that stirs the leaves, nature's harmony mirrors the delicate equilibrium that enzymes maintain in their own realms. As we continue to understand the nuances of temperature and pH, we stand at the precipice of new discoveries. The potential for innovations in medicine, food production, and biotechnology lies within our grasp. Each adjustment we make, whether in a laboratory or an industrial setting, has the power to unlock new pathways, leading to advancements that can enhance our quality of life. So, what lies ahead in this journey of exploration? The answers are as vast as the cosmos, waiting to be uncovered. Each experiment, every observation, brings us closer to a greater understanding of the world around us. In this intricate dance of science and nature, we are reminded of our place within the grand tapestry of life, where every temperature change and pH adjustment plays a role in the unfolding story of existence. As we continue to navigate this fascinating landscape, we embrace the wisdom of those who came before us, while looking forward to a future filled with discovery. The impact of temperature and pH on enzyme function is merely the beginning of a deeper understanding of the biochemical symphony that sustains life in all its forms. The intricate interplay between temperature and pH not only shapes the activity of enzymes, but also holds the key to unlocking potential innovations in various industries. When we consider enzyme kinetics, it becomes evident that understanding these factors allows us to tailor processes that maximize efficiency. For instance, in the textile industry, enzymes are employed to modify fabrics, enhancing their texture and durability. Imagine the precision with which enzymes like cellulases work to break down cellulose fibers, creating smoother and softer materials without the harsh chemicals traditionally used. This eco-friendly approach reflects the growing trend toward sustainable practices, marrying the art of fabric production with the science of enzymology. Temperature also plays a critical role in the production of biofuels, where enzymes are harnessed to convert biomass into energy. Picture a vast field of corn, ready to be transformed into ethanol. The enzymatic breakdown of starch into sugars is a pivotal step in this process. By carefully controlling the temperature during this conversion, producers can optimize the yield of fermentable sugars. This meticulous attention to detail ensures that the energy extracted from these plants is maximized, offering a renewable source of fuel that aligns with the global shift toward sustainable energy solutions. Moreover, the food industry benefits immensely from the knowledge of enzyme behavior under different pH levels. Take the process of brewing, for example. The flavor profile of beer is a delicate balance, influenced by the pH at various stages of fermentation. Imagine brewers as chefs, skillfully adjusting the acidity of their mixtures to coax out the desired flavors. The enzymes involved in this process convert starches into sugars and subsequently into alcohol, all while the pH remains finely tuned to create that perfect brew. The application of this knowledge extends to the dairy industry as well. The delicate balance of temperature and pH during yogurt production is crucial. The introduction of specific bacterial cultures initiates a fermentation process where enzymes thrive, transforming milk into a creamy delight. The temperature must be carefully regulated to ensure that the enzymes remain active, while the pH shifts as fermentation progresses, locking in the signature tartness that yogurt lovers cherish. As we consider these applications, it is worth pondering the broader implications of enzyme optimization. How can we further harness the potential of enzymes to address the challenges of our time? The answer lies in ongoing research and innovation. Scientists are continually exploring new enzyme variants through techniques such as directed evolution, which mimics natural selection in the laboratory. By exposing enzymes to various conditions and selecting those that exhibit improved performance, researchers are crafting enzymes that can withstand extreme temperatures and pH levels, broadening their applicability in diverse industrial settings. This research echoes the pioneering work of scientists like Frances Arnold, who once stated, "Nature is the best chemist." Her insights into the design of enzymes have opened doors to advancements that extend far beyond traditional applications. Imagine engineers harnessing these specially designed enzymes to tackle pollution, breaking down plastics in our oceans, or converting waste into valuable resources. The possibilities seem endless, a horizon filled with the promise of biocatalysis as we strive for a more sustainable future. Yet, even as we contemplate these advancements, we must remain mindful of the delicate balance that governs enzyme activity. This balance is not merely a scientific principle. It is a reflection of the interconnectedness of all living systems. Just as enzymes require specific conditions to perform their roles, we too thrive when our environments are nurtured. The air we breathe, the water we drink, and the ecosystems we inhabit are all part of the larger picture, reminding us that our well-being is intertwined with the health of our planet. As we deepen our understanding of enzyme function, we find ourselves embarking on a journey that transcends the laboratory. Each discovery, each innovation, is a step toward a future where science and nature coexist harmoniously. The impact of temperature and pH on enzyme function serves as both a reminder of the beauty of life's intricacies and a call to action to preserve and enhance the world around us. In this quest for knowledge, we uncover the stories of those who have dedicated their lives to studying enzymes and their remarkable capabilities. From the early biochemists who first illuminated the pathways of metabolism to the modern-day innovators pushing the boundaries of biotechnology, these individuals have shaped our understanding of life at the molecular level. Their work inspires us to continue exploring, to delve into the unknown, and to embrace the challenges and opportunities that lie ahead. As we stand at this crossroads of discovery, one cannot help but wonder, what new insights await us in the realm of enzyme research? The potential for breakthroughs in medicine, agriculture, and environmental science beckons us forward. Each experiment conducted, each hypothesis tested, is a testament to human curiosity and resilience. The journey of enzymes is a story of transformation, an ongoing narrative woven into the fabric of life itself. With each passing moment, as we uncover the layers of complexity that govern enzyme function, we become more attuned to the rhythms of nature. Just as enzymes catalyze the reactions that sustain life, our exploration of their capabilities can lead to a brighter, more sustainable future for all. In this light, the understanding of temperature and pH is not just a scientific endeavor. It is an invitation to engage with the world around us, to appreciate the delicate balances that sustain life, and to seek pathways that enhance our collective well-being. The story of enzymes is far from over. It continues to unfold, inviting us to participate in the exploration of the wonders that lie within the microscopic realm, where life's chemistry dances in the shadows of our understanding. Chapter 10: Future Directions in Enzyme Research. The landscape of enzyme research is evolving rapidly, driven by emerging technologies that promise to unlock new potentials. As we look ahead, we see the dawn of innovative techniques that enhance our ability to modify and engineer enzymes, taking us beyond the natural capabilities of these remarkable proteins. One such technology is directed evolution, a method that mimics natural selection in the laboratory. Imagine a vast field of flowers where only the most vibrant and resilient bloom. In a similar fashion, directed evolution allows scientists to create a diverse library of enzyme variants, subjecting them to selective pressures that reveal the most effective forms. This process can lead to enzymes with enhanced stability, specificity, or catalytic efficiency, vastly expanding their applications. Another groundbreaking approach is the use of computer-aided design and artificial intelligence in enzyme engineering. Picture a skilled craftsman meticulously shaping a piece of wood into a beautiful sculpture. With the aid of advanced algorithms, researchers can now predict how changes in an enzyme's amino acid sequence will impact its function. This not only accelerates the design process, but also allows for the creation of enzymes with entirely new functionalities, tailored to meet specific needs. Moreover, the advent of synthetic biology has opened new doors for enzyme research. By integrating biological components with engineering principles, scientists are crafting entirely new metabolic pathways, often incorporating enzymes from diverse organisms. This interdisciplinary approach holds the potential to revolutionize industries, from pharmaceuticals to agriculture, by enabling the production of complex molecules in a more sustainable manner. In this context, the potential for enzymes to contribute to sustainable practices is immense. Enzymes play a crucial role in biocatalysis, where they facilitate chemical reactions under mild conditions, offering an environmentally friendly alternative to traditional chemical processes. Imagine a factory where instead of harsh chemicals and extreme temperatures, gentle enzymes operate in a serene environment, effectively converting raw materials into valuable products while minimizing waste. This shift towards biocatalysis not only reduces energy consumption, but also decreases the ecological footprint of industrial processes. Consider the impact of enzymes in the production of biofuels. By utilizing enzymes to break down plant biomass into fermentable sugars, we can harness the energy stored in renewable resources, contributing to a more sustainable energy landscape. The application of enzymes in producing bioplastics further exemplifies this potential. Enzymatic processes can transform renewable biomass into biodegradable materials, reducing our reliance on fossil fuels and mitigating plastic pollution. Yet, as we explore these promising avenues, challenges and opportunities arise in enzyme-related research. One significant challenge is the stability and activity of enzymes in harsh industrial conditions. While enzymes are fine-tuned by nature for optimal performance within living organisms, they often struggle to maintain their function outside of these environments. Researchers are diligently working to enhance enzyme stability through various strategies, including mutagenesis and encapsulation techniques. Imagine these enzymes as delicate flowers, needing protection from the elements while still performing their vital roles. Another hurdle is the cost associated with enzyme production and purification. While enzymes can offer significant benefits, the economic feasibility of large-scale production remains a concern. Advancing production methods and optimizing fermentation processes can help address these issues, making enzymes more accessible for widespread applications. Moreover, as we venture into the realm of personalized medicine, the role of enzymes in diagnostics and therapeutics is gaining prominence. Imagine a world where enzyme-based tests can provide rapid and accurate health assessments, tailoring treatments to individual needs. This is not merely a dream. It is a tangible future. Enzymes can be harnessed to develop biosensors that detect disease markers with remarkable precision, paving the way for early diagnosis and improved patient outcomes. As we embrace the future of enzyme research, collaboration across disciplines becomes essential. Biochemists, engineers, and environmental scientists must come together, pooling their expertise to unlock the full potential of enzymes. This collaborative spirit will drive innovation, allowing us to tackle the pressing challenges of our time, from climate change to public health. Each step we take into this future is illuminated by the promise of enzymes. Consider their role in the circular economy, where waste is repurposed into valuable resources. Enzymes can be utilized to break down organic waste, converting it into biogas or biofertilizers, thereby reducing landfill dependency and enhancing soil health. The future of enzyme research is indeed bright. With emerging technologies and sustainable practices at the forefront, we stand on the brink of a new era, where the potential of enzymes to transform our world is limited only by our imagination. As we journey forward, may we embrace the opportunities that lie ahead, fostering a deeper understanding of these biological wonders and their role in shaping the future of our planet. The potential of enzymes to drive sustainable practices extends beyond mere applications. It encompasses a profound transformation in how we perceive and utilize resources. Enzymes are integral to biotechnological advances that aim to minimize our ecological footprint. For instance, the role of enzymes in waste management is becoming increasingly significant. Imagine a world where organic waste does not simply accumulate in landfills, but is instead efficiently converted into valuable products through enzymatic processes. This is the promise of enzyme technology, where waste can be transformed into biofuels, compost, or even animal feed. Enzymes like cellulases and lignases can break down cellulose in plant materials, enabling the conversion of agricultural residues into fermentable sugars. These sugars can then be fermented to produce ethanol, a renewable energy source. Picture farmers collecting leftover crop materials, which would otherwise be discarded, and instead channeling them into a bioprocess that generates energy, fostering both economic and environmental benefits. This illustrates how enzymes can contribute to a circular economy, where resources are reused, reducing demand for virgin materials. The textile industry, notorious for its environmental impact, is also witnessing a transformation through enzyme technology. Enzymes can be employed to replace harsh chemicals traditionally used in processes such as bleaching and dyeing. Imagine vibrant fabrics emerging from a gentle wash with enzymes, preserving their colors and textures while minimizing water usage and chemical pollution. Such innovations not only enhance the sustainability of textile production, but also align with consumer demand for eco-friendly practices. However, as we delve into these sustainable applications, we must also acknowledge the challenges that accompany this progress. One pressing concern is the need for enzymes that can function efficiently under the specific conditions required in various industrial processes. Each application may present unique challenges, such as high temperatures, varying pH levels, or the presence of inhibitors that can affect enzyme activity. The quest for robust enzymes capable of withstanding these conditions drives ongoing research and innovation. Another significant challenge lies in the scalability of enzyme production. While laboratory-scale enzyme applications demonstrate remarkable potential, translating this success to industrial-scale processes often presents hurdles. The economic viability of enzymes hinges on the development of cost-effective production methods. Advances in fermentation technology and genetic engineering can enhance yield and reduce production costs, allowing us to harness the full capabilities of enzymes in sustainable practices. In addition to these challenges, the realm of enzyme research is brimming with opportunities. As we witness the convergence of biotechnology and synthetic biology, the potential to design custom enzymes tailored for specific applications expands exponentially. Imagine designing an enzyme that can efficiently break down plastics into harmless byproducts, addressing one of the most pressing environmental issues of our time. This vision is not far-fetched. Researchers are already exploring ways to engineer enzymes that can tackle pollutants and transform waste materials into useful resources. Moreover, the integration of enzymes into the field of personalized medicine highlights another exciting frontier. Enzymes can be utilized to create targeted therapies, where specific enzymes are designed to interact with particular biological pathways. This precision holds the promise of more effective treatments, tailored to individual patient needs. Imagine a healthcare system where enzymatic therapies are not only more effective, but also safer, minimizing side effects by targeting only the necessary biological processes. As we stand at this juncture of discovery, the road ahead is illuminated by the collaborative efforts of researchers, engineers, and environmentalists. The synergy between these disciplines fosters a rich environment for innovation, allowing us to explore new horizons in enzyme research. By sharing knowledge and expertise, we can craft solutions that address global challenges, from climate change to healthcare disparities. Consider the role of enzymes in food security, an issue of paramount importance as our global population continues to grow. Enzymes can facilitate the production of nutritious food sources, enhancing the digestibility of grains and legumes through enzymatic treatments. Imagine communities benefiting from fortified foods, where enzymes enhance nutrient availability, supporting health and wellness. The challenges we face in enzyme-related research are not roadblocks. They are invitations to explore deeper into the science of life. Each challenge presents an opportunity for breakthroughs that could redefine our relationship with the environment and with our health. As we look to the future, the potential of enzymes seems boundless. The intricate dance between nature and technology encourages us to envision a world where enzymes play a central role in sustainable practices, not just as catalysts for chemical reactions, but as pillars of a new era of innovation. With every discovery and breakthrough, we inch closer to the realization of a future where the harmony of enzymes and nature paves the way for a healthier planet and society. In this ever-evolving landscape, it is crucial to remain open to new ideas and approaches. The future of enzyme research is not solely about scientific advancements. It is also about understanding the ethical implications of our work and striving for solutions that benefit all of humanity. Each enzyme discovered, each application developed, carries the weight of responsibility toward our planet and future generations. So, as we continue to explore the vast potential of enzymes, let us do so with a sense of wonder and purpose. Enzymes are not merely tools of science. They represent a bridge to a sustainable future, linking innovation with responsibility. The journey ahead may be complex, but with each step, we are reminded of the extraordinary possibilities that lie within the microscopic world of these biological catalysts. As we envision a future rich with possibilities, the intersection of enzyme research and sustainability becomes increasingly clear. Enzymes not only hold the promise of transforming industrial practices, but also of reshaping our relationship with the environment. The potential to harness enzymes for bioremediation, for example, is a captivating frontier. Imagine a landscape marred by pollution, where toxic substances linger in the soil and water. Enzymes can be engineered to break down these harmful compounds, converting them into harmless byproducts. This application highlights enzymes as environmental stewards, capable of restoring balance to ecosystems while promoting human health. The exploration of enzyme applications in agriculture is equally compelling. Enzymes can be utilized to enhance soil health, promoting the availability of nutrients to plants. Picture a thriving farm where enzymatic treatments improve the breakdown of organic matter, enriching the soil and supporting robust crop growth. This not only boosts agricultural productivity, but also aligns with sustainable practices that prioritize ecological balance. Furthermore, enzymes can assist in developing biopesticides, offering a gentler alternative to chemical pesticides that often harm beneficial organisms. As we venture into these uncharted territories, it is essential to recognize the challenges that accompany such advancements. The complexity of enzyme interactions within diverse environmental contexts can pose significant hurdles. For instance, the performance of enzymes in soils or aquatic systems can be influenced by numerous factors, including temperature fluctuations and varying pH levels. Researchers are tasked with understanding these dynamics to optimize enzyme functionality in real-world applications. In this landscape of challenges lies an array of opportunities. The ongoing advancements in genetic engineering and synthetic biology empower scientists to tailor enzymes for specific environmental conditions. Imagine the potential of a custom-designed enzyme, perfectly suited to thrive in a particular ecosystem, breaking down pollutants with unprecedented efficiency. This vision is becoming increasingly attainable as our understanding of enzyme structure and function deepens. Collaboration will be paramount in addressing these challenges. As interdisciplinary teams of biochemists, ecologists, and engineers come together, their collective expertise can drive innovation and develop robust solutions. By sharing knowledge and resources, these collaborations can lead to groundbreaking discoveries that amplify the impact of enzyme research on sustainability. Furthermore, the ethical implications of enzyme applications warrant thoughtful consideration. As we harness the power of enzymes to address environmental issues, we must ensure that these technologies are developed responsibly and equitably. The impact of enzyme-based solutions should be evaluated not only in terms of efficacy, but also in their potential effects on local communities and ecosystems. In the realm of enzyme research, the future is vast and nuanced, filled with the promise of discovery and transformation. Each breakthrough we achieve enhances our understanding of these remarkable biological catalysts and their potential to influence our world positively. As we continue to explore their capabilities, we should remain guided by a vision of sustainability, equity, and respect for the intricate web of life that surrounds us. Consider the role enzymes play as we navigate the challenges of resource scarcity and climate change. In this context, enzymes are not just tools, they are vital components of a larger strategy aimed at fostering resilience in our ecosystems. Enzymatic processes can enhance carbon sequestration in soils, which not only mitigates climate change, but also supports agricultural sustainability. Picture a farm where healthy soils capture carbon, nourishing crops while contributing to the fight against global warming. The dialogue surrounding enzyme research is shifting, reflecting a growing awareness of the interconnectedness of science, technology, and society. As we embrace this shift, we must cultivate a culture of curiosity and collaboration, where diverse perspectives contribute to the evolution of enzyme applications. By engaging with communities, policymakers, and industry leaders, scientists can ensure that their research translates into meaningful solutions that resonate with societal needs. In the coming years, the synergy between emerging technologies and enzyme research will likely pave the way for unprecedented innovations. The integration of artificial intelligence in enzyme discovery and optimization will enhance our ability to identify promising candidates more efficiently. Imagine a future where AI algorithms sift through vast databases of enzyme sequences, predicting their potential functions and guiding researchers toward the most promising leads. This convergence of biology and technology is poised to accelerate advancements in ways we are only beginning to comprehend. As we peer into the horizon of enzyme research, we envision a landscape filled with potential breakthroughs, an evolving narrative that intertwines scientific discovery with the pursuit of sustainability and health. The future beckons with the promise of enzymes, inviting us to unlock their secrets and harness their power for the greater good. With each step we take, we reaffirm our commitment to advancing knowledge, fostering collaboration, and creating a world where the potential of enzymes can be fully realized.